Morning, on. I'm joining through my cell phone this morning from Ottawa. I'm the country representative for ICA in Canada and the Secretariat for Puerto Norte. This is day 2 of a 2-day workshop. We do this every year. Some years are hybrid, some years are in person in Mexico. And this, we're delighted to focus on more of a bird flu and screwworm and other emerging diseases today. For those of you who were there yesterday, welcome back. For those of you who are new, I was laughing earlier because I was looking at my list of, usually when I do housekeeping, I'm like, oh, the Wi-Fi and the bathrooms, but you know where those are. So that's all good. The task force, so we've got at Puerto Norte multiple task forces. For those of you who don't know us, it's one of ICA's technical cooperation mechanisms, about 7 at ICA. We are responsible for the North, Canada, US, Mexico, the Caribbean. We have other task forces on plant health and genetics and resilience. This one on animal health is led by Dr. Susana Flores at INIFAP in Mexico City. And we're joined by Dr. Roxanne Motroni from the US, ARS at the USDA, and Dr. Una Magala in Winnipeg at the CFIA. So it's a great team. We've worked with them a long time. And they've put together a wonderful agenda for you today. Same as yesterday, it runs to about 2:30, a hard stop. And Dr. Magala will be facilitating today as opposed to Dr. Motroni yesterday. And ideally, there's some time for dialogue and Q&A after each presentation. If there isn't, we will switch to chat. That doesn't mean you can't chat throughout the entire day. So if you have a question for a particular scientist presentation, feel free right away to monitor the chat and submit your questions in during the session or after. And the scientists will help with responding to you. The Camino in Mexico City, our colleague, is managing the Puerto Norte is our coordinator. She's some of the questions we had yesterday in terms of videos and all that, and she can answer in the chat, and it'll be online on YouTube through our PuertoNorte.net website. And so I would like to invite the head of the task force to say a few opening remarks for this morning session, and I'll switch it over to Dr. Susana Flores at INIFAP. Over to you.
Hola, buenos días. Muchas gracias, Jaycee. Bueno, es para mí un honor darle la más cordial bienvenida a todos ustedes a este taller de la Fuerza de Trabajo de Salud Animal de Puerto Norte. Como bien dijo Jayce, doctor Roxanne y doctora Luna, creamos este programa de trabajo de 2 días, en donde el día de ayer hablamos un poco más de los aspectos que afectan la salud animal, pero bajo el contexto de una salud. Hablamos de temas de resistencia antimicrobiana, cambio climático, algunas enfermedades transmitidas por vectores. Y el día de hoy vamos a enfocarnos específicamente a dos enfermedades emergentes de gran relevancia, que es influenza aviar y gusano barrenador del ganado, ¿no? Igualmente, como el día de ayer, los invitamos a formular sus preguntas para los panelistas al término de cada una de las presentaciones, o bien como lo dijo Jayce en el chat. Y esto es con el objetivo de, obviamente, intercambiar experiencias, conocimientos y, e información, ¿no? Porque al final ese es el objetivo principal de Procenorte, ¿no? Tratar de crear una comunidad de colaboración trinacional para encontrar soluciones de forma conjunta. Yo les agradezco enormemente a nuestros panelistas su disposición para compartirnos sus experiencias y sus conocimientos, y a ustedes por escucharnos el día de hoy. Entonces, sin más preámbulo, le doy la palabra al doctor Aruna para que nos haga el favor de comenzar con nuestro taller. Muchísimas gracias.
Thanks, Susanna. Thanks, JC, for the introduction. I'm Aruna Ambagala. I'm the head of the Mammalian Diseases Unit here at the CFIA, National Center for Foreign Animal Disease, and the WOHA Reference Laboratory for African Swine Fever and Classical Swine Fever. So welcome, everyone, and good morning. And so we have 4 presenters in the morning, and then we have a health break, and then we have 2 presentations in the afternoon. And each person has 30 minutes, and I appreciate if you stick to 30 minutes. And if additional time we have, we will use that for questions. So our first speaker today, Dr. Anthony Signore from Canadian Food Inspection Agency, and he will talk about surveillance and phylogenomics of clade 2.3.4.4b viruses, the emergence of genotype D1.1 and oseltamivir resistance. Sorry if I messed up your title, Anthony.
Okay, that's quite all right. Thank you very much for the introduction. And thank you very much to the organizers for giving me the opportunity to present here today. I'm going to use this opportunity to tell you a little bit about 2 different studies that we've done here at the CFIA's National Center for Foreign Animal Disease. But before I jump right into that, because this is a diverse audience, I want to give you just a very brief background on influenza A viruses. And you can use this going forward throughout today for the other lovely influenza talks that we're going to see. So they're a family of viruses that belong to the Orthomyxoviridae family. It is an envelope virus. It has 8 segments of single-stranded RNA as its genome. And as you can see from this textbook image here, there are a number of proteins dotted outside the virion. Those proteins are primarily hemagglutinin, or HA, and neuraminidase, NA. These proteins are primarily involved in cell entry and release. They are also the primary immunogenic proteins, and they are also how we subtype these different viruses. So there are 16 HA subtypes and 9 NA subtypes that primarily circulate in wild birds. And depending on the complement of the HA and NAs on the surface of the virus, that's how we get the different subtypes like H3N2, H5N1 that you may have heard of, etc. So these viruses, they primarily circulate in anseriforms. These are geese, ducks, and swans, and tetraforms, shorebirds. But they can infect a number of viruses, including most other birds, mammals, and including humans. There was a highly pathogenic strain of avian influenza virus, you might see throughout the talk, referred to as HPAI, of subtype H5N1 that was circulating in Western Europe in 2021. In late 2021, this virus diffused across the Atlantic Ocean and found its way to North America. The first detection of this particular virus in North America was in a blackback gull in St. John's, Newfoundland, Canada. And this led to an outbreak that has impacted countless wildlife species, millions of poultry, and as well as non-avian livestock, as I think we're going to hear about later today. And we assessed the impact of this outbreak in Canada, in Canadian wildlife, with an enormous interagency surveillance effort. So before I move any further, I just want to take a moment to acknowledge Canada's interagency surveillance program for avian influenza viruses. It is a huge collaborative effort, and they have been very busy over the last few years. Almost 60,000 live, dead, and sick animals, wildlife specimens have been tested, and just under 10% come back as positive for highly pathogenic H5 avian influenza viruses. These viruses have frequently spilled over onto poultry farms. The CFIA's National Emergency Operations Center for HPAI does surveillance on poultry farms, and they have found that almost 600 farms have been infected with this virus, impacting more than 14 million birds. And so all of these positive samples, whether they're in a poultry farm or in wildlife, the vast majority of them end up here at Canada's National Centre for Foreign Animal Disease, where they undergo confirmatory testing and whole genome sequencing. One of the most remarkable things that we've found from our whole genome sequencing is the frequency with which these viruses reassort with low pathogenic North American viruses. So reassortment is when two viruses co-infect the same cell. During their genome replication process, their genome segments can get jumbled up together and repackaged into different genomic constellations, creating a new reassortment virus that is a combination of the two parents. In Canada and the United States, these H5N1 viruses that landed here from Europe reassort frequently with North American low pathogenic viruses, and they have created more than 100 genotypes that have been detected in Canada and the United States. Each node in the network that you're seeing here represents a unique H5N1 reassortment that has been detected just in Canada alone. The size of each of these nodes is proportional to its detection frequency, so you can see that it varies depending on the reassortment. And the arrows represent the reassortment event that created the progeny virus from the parental reassortment. So it represents the ancestry of each one of these reassortments. And just like other influenza outbreaks or other pathogen outbreaks in general, the proportion of these viruses, the frequency in which they're detected, changes over time. It also changes over space. So this map here you're seeing up in the corner represents the spread of the genotype A1. This is the virus that came here from Western Europe that was initially found in that blackback gull that I highlighted earlier. It spread down the Atlantic coast of North America in the fall of 2021 and the winter of 2022. And then subsequently in the spring of 2022, migrating birds brought it back up north through the Midwest United States, and it made some inroads into Western Canada. However, it was very sparsely detected in Western Canada because it was replaced, both temporally and spatially, by a different reassortment virus. So genotype B2.1 emerged right in the center of Canada and the United States. It outcompeted genotype A1, replaced it, and took over this geographic area, but didn't spread much further than this, because this virus was eventually replaced by a different genotype, B3.2. The dynamics of this one is a little difficult to sum up in a still image, so I have a video to supplement. This virus emerged in the same place as B2.1, the very center of Canada and the United States. It replaced B2.1, outcompeted it, and became the dominant virus in this region in the time leading up to the fall southward migration. In that time, birds migrating south moved the virus throughout the United States, Mexico, South America, and eventually made inroads into Antarctica. This was and remains to be one of the most successful viruses that we've seen in this current outbreak, both in terms of the number in times it's been detected, but also the geographic distance that it's covered. But despite all of the success of this virus, it too, the following season itself, was replaced by another reassortment. B3.6 replaced this virus in much the same fashion that B3.2 replaced B2.1, and going back so far, it emerged again right in the center of Canada and the United States, outcompeted B3.2, replaced it in the region, and then was spread by southward moving migrating birds. So what we're seeing is a huge amount of genetic variation that emerges via reassortment, which selection can then act upon. More fit variants emerge, they replace the previously dominant viruses and spread throughout the continent. But what's interesting is that despite all of this genetic variation that we're seeing, the most successful variants that I just highlighted, they're all direct descendants of one another. So we see an unbroken chain of reassortment. A1 reassorted to create B2.1, which replaced it. B2.1 reassorted to create B3.2, which replaced it, and so on with B3.6. So these successive reassortment events seem to be yielding compounding fitness increases. And if we estimate the relative fitness by this top line here, we see the fitness does successfully increase from A1, B2.1, B3.2, and B3.6. Similar things, based on a preprint that was just published a few weeks ago, have been found in Europe, where reassortment of these internal segments is creating increasing fitness gains. This particular study, although it is still a preprint, but they did find, just based on reassortment, they found differences in viral growth kinetics, differences in polymerase activity, as well as differences in the shutoff of host gene expression. So we're seeing, and this is again the North American example, a chain of successive reassortment events. They're only introducing new internal segments. And so what I mean by that, generally the fitness of these viruses is thought to be tied very tightly to the external proteins, the hemagglutinin and neuraminidase that I mentioned earlier. But we're seeing reassortment events that only introduce new internal segments, each apparently giving rise to a more fit virus that supplanted its parent. And this chain continued throughout the first 3 years of the outbreak, unbroken, up until the fall of 2024, when North America saw something that it hadn't seen before, at least in terms of this outbreak. Up until September 2024, all of the reassortment events that we've seen, all of those more than 100 that I identified a few slides ago, they did not contain any of the surface proteins. So no hemagglutinin, no neuraminidase, or the M2 protein. So it hadn't represented any sort of antigenic shift until September 2021, when a new genotype was detected, which eventually became named genotype D1.1. If we look at a phylogenetic tree of just the hemagglutinin segment, and a second phylogenetic tree of just the neuraminidase segment, we see these D1.1, we see some topological differences. The H5 clusters with the highly pathogenic viruses that have been the sole cause or the heart of this outbreak, whereas the neuraminidase clusters with low pathogenic N1 viruses. So this is the first instance we've seen of a reassortment event that has changed one of the surface proteins. This was first detected in Alaska in September 26, 2024. The first Canadian detection happened very shortly after. It was detected by the BC CDC on October 6, 2024. And this virus very quickly not only replaced all other circulating or most other circulating viruses, but spread very quickly across the Americas. You can see just from this plot here, the sheer numbers, it dwarfs basically anything that we've seen before. And if we look at the spread, it was first detected at the very end of September 2024. It had already reached Southern California just a few weeks later. So it spread very quickly south. It also spread very quickly west to east as it reached the Atlantic flyway by early November. So not only was this unprecedented in its genomic makeup, but it was unprecedented in the speed and distance it covered in such a short amount of time. But one thing I want to highlight, another thing that we hadn't seen before, is a very particular mutation, a mutation that confers drug resistance. So viruses collected on a poultry farm near Abbotsford, BC on November 1st, 2024 revealed an exceedingly rare amino acid substitution. It was found previously, before this, in only 8 of almost 18,000 isolates that had been sequenced across the world, with no evidence of forward transmission previous to this detection. This particular substitution in the neuraminidase gene, where a histidine is replaced with a tyrosine, it confers strong resistance to the neuraminidase inhibitor oseltamivir, also marketed as Tamiflu. Tamiflu is the most commonly used prophylactic and treatment for human influenza infections. This particular substitution emerged in a poultry farm in British Columbia and quickly spread across 12 poultry farms in the province. Fortunately, however, it has not detected since November 30th, 2024, and critically, it is not detected in any wildlife samples. So what you're seeing here is a molecular model of the mechanism by which it confers oseltamivir resistance. So down here in the bottom left corner is indeed the oseltamivir molecule. This is the drug that inhibits the neuraminidase protein for the treatment of influenza infections. We can see the replacement of histidine by a larger tyrosine residue slightly pushes on the amino acid at position 277, which forces oseltamivir out of its binding pocket. This is a very well-studied substitution. However, it has never been studied on these particular genetic backgrounds on this particular H5N1 virus. This has all been done on other largely human-based viruses. So here at the NCFAD and with our partners at the Public Health Agency of Canada, we tested if this particular substitution does indeed confer resistance to this influenza drug. And yes, indeed it does. It does confer the same resistance to this drug as we've seen in other viruses. But what's interesting is that this substitution emerged at all. This substitution typically emerges from the administration of oseltamivir or Tamiflu. Administration of Tamiflu provides some selection pressure for this drug resistance mutation to emerge, and then it does. As far as we're aware, there's been no Tamiflu use on this poultry farm where these viruses emerged. And what's more interesting is that the virus continued to spread after it did spontaneously emerge, because this particular substitution is generally seen as less fit than the wild type. This is largely because the tyrosine at position 275 reduces the stability of the neuraminidase protein relative to the wild type, which has a more stable neuraminidase with a histidine at position 275. We think this may be due to permissive mutations that increase the overall stability of the neuraminidase proteins that are found in these D1.1 viruses. There are 4 that have been previously identified in H1N1 proteins, and one in particular that is not found in any H5N1 viruses that we've seen throughout this outbreak. So it could be these 4 permissive mutations that are negating the fitness impact of this substitution at position 275. However, once again, just like when testing the oseltamivir resistance, these permissive mutations were identified on a much different genetic background. They were done so on human H1N1 viruses. We don't know if they have the same phenotypic effect on these H5N1 viruses. So in an effort to determine if these genotype D1.1 viruses may be pre-adapted to carrying these oseltamivir resistance markers, we use some genomics and some in silico prediction tools to do this. So what you're looking at here is effectively a tree of all clade 2344B H5N1 viruses that have been detected throughout this outbreak based on just their neuraminidase proteins. And you can see that these D1.1 viruses that I highlighted, they cluster in a completely different clade than the other 2344B viruses because, as I mentioned, they have a North American-derived neuraminidase protein. They have a different ancestral lineage than the others. From this tree, we can estimate the ancestral protein sequence at every single node. From those protein sequences, we can create molecular models for both the ancestor of other D2344B viruses, as well as the ancestor of all D1.1 viruses. From these molecular models, both at the terminal ends of the tree and at every single node, we can estimate how inherently stable these proteins are. So we use an estimate called delta G. It's the energy of unfolding. So essentially, the more negative value means more energy is required to unfold the protein, which means it is more inherently stable. And as you can see from these numbers under the very small molecular models that I produced for you here, that the D1.1 proteins are inherently more stable than other clade 2344B viruses. And so the next step here is to identify which particular amino acids are contributing to these differences in stability and which are allowing these oseltamivir resistant mutations to emerge and potentially spread across the Americas. And so to do that, we can get statistical support for every amino acid position that may affect the change in protein stability that we're seeing all across this phylogenetic tree. And we can map the net effect of each substitution and how much it actually impacts the stability of the protein. So I just want to highlight one up here. So this particular substitution at position 84 in the neuraminidase chain has a very high probability of impacting the stability of the protein. If we look at its estimated effect, it's estimated to change about 2 kilojoules per mole, which is most of the difference we see between D1.1 proteins and the other H5N1 proteins. So using methods like this with rapid genomic surveillance as well as in silico tools, we can identify concerning mutations before they have a chance to spread and potentially mitigating impacts to pandemic potential viruses. And so with that, I have a lot of people that I want to thank for this work. This is primarily 2 different studies, which includes a lot of people. The individual names you're seeing here are just the co-authors on these studies. Again, I want to acknowledge Canada's Interagency Surveillance Program for avian influenza viruses, as well as a number of other units that have contributed both time and research to this project, the NCFAD Avian Diseases Unit, our Genomics Unit, our National Emergency Operations Center, as well as Sage News Hospital, the USDA National Vet Services Lab, as well as the Public Health Agency of Canada. And then I, of course, want to thank all of you for your attention, and we have some time for questions. I'll be happy to take those if you have any.
Thanks, Anthony. Thanks for the excellent presentation and all the information you shared with us. Are there any questions? I don't see any questions in the chat. Oh, Rahat has a question. Rahat, go ahead.
Hi, Anthony. It's an excellent talk. I really enjoyed it. Thank you so much. I have a question about the replacement and reassortment. Is there a correlation between this reassortment with the passages? If, for example, if one virus is more successful in infecting multiple hosts or passing through multiple hosts, is there a correlation that there is more reassortment? I have a couple of questions.
Okay,
Oh,
yeah. I haven't quite, at least not with statistical identified which hosts significantly contribute to reassortment, but there are definitely differences in the way these viruses spread and the way they affect different hosts. So just in particular, just changing a few of the internal segments does impact their survivability. Those are a few things that we have actually tested in the lab. Some viruses are much less survivable by just changing one of the internal segments than the other. So there are certainly host differences tied to fitness, but as far as the effect of and the frequency of reassortment in a particular host, we don't actually have those numbers at this time. It seems to be most of these reassortments have emergent answeriforms. All of them, except all the ones I identified, all of them have emergent answeriforms, so geese and ducks, with the exception of one.
Okay, thank you. I just have one more question. So are you seeing any correlation between fitness and virulence? That if there is if the fit virus has less virulence, for example?
Yeah. Let me just back up a couple of slides. I can give you an example. So the short answer is yes. So if we look here, these 2 viruses, B1.2 and B1.3, I didn't talk about them much because they didn't have a lot of impact on the overall outbreak in North America. They were isolated to a fairly small genomic region, but one replaced the other in that region. This one here, B1.3, is the most virulent virus that we've tested in our lab, and it is indeed estimated to be more fit than its predecessor, B1.2. So yes, in this particular case, heightened fitness is actually associated with higher virulence, but that's not always the case.
Thank you.
Okay, thanks. So we have a question. What does the level of genomic plasticity say about the likelihood of vaccines being HPAI?
Well, so all of these viruses that you're seeing in this network, plus all of the other reassortments that have taken place outside of Canada and the United States, they all carry the same hemagglutinin gene, which is the target of vaccines. So they should largely be unaffected by any vaccination efforts if that vaccination effort is targeting the hemagglutinin segment.
Thank you. Anthony, okay, let me see whether there are any questions. If not, I have a question for you. So you talk about the drug resistance strains that you talk about, and do they carry any mammalian adaptation markers in those viruses?
Well, in the few that carry the drug resistance marker, no, we haven't seen any mammalian adaptations, which again is a good sign. in addition to it being stamped out quickly. A few mammalian markers have emerged in D1.1 viruses in general, but none that co-carry both the mammalian adaptations as well as the oseltamivir resistance.
Excellent. One more question for you. I mean, you talk about the virulence. I'm sure these viruses show high fitness and also have probably the amount of shedding is probably higher and then transmission is higher as well, I guess, compared to others in your studies.
Yeah, they certainly differ based on genotype. We don't have sort of the complete data set to really test the difference between all of these viruses that I highlighted here, but there are absolutely differences between shedding and virulence between the different genotypes, which you'll hear a little bit more from Tamaru next, I believe.
Excellent. Are there any questions? I don't see any questions. Susanna, do you see anything in the chat for in Spanish? I don't see anything. So hope.
No, I can't see anything there.
Perfect. Okay. Oh,
There is one more
there's
question.
another question from Rahat. Rahat, go ahead.
Oh, sorry.
So
It was just about the
comment.
tool. Yeah, comment that we can
yeah.
talk maybe later. Yes.
Yeah, happy to discuss. It's not really a tool. It's more of a series of tools that I smashed together for my purposes, but happy to chat about that later with you if you'd like.
Excellent. Okay. I think we can move to the next talk. Dr. Tamaru Aliki from Canadian Food Inspection Agency will talk about pathogenicity and transmission of avian influenza virus in small ruminants. Tamaru, you can share the slide. Maybe you can go ahead and press the presentation view. And I think you need to share the other screen, like perfect. Thank you. Tamaru, you can start. We cannot hear you, Tamaru.
Okay, yeah. Okay, can you hear me now?
Yes.
Okay, so yeah, I thank you all for organizing this meeting. And I am presenting more of our data, which we collected from experimental studies. And the topic of my presentation is on pathogenicity and transmission of H5N1 and also H5N5 in small ruminants. Yeah, this slide is to show you, I don't want to repeat it, this is to show you about the panzootic states of this clade 2.344b virus. As you can see, this virus was emerged in Europe, in Netherlands, and then spread to North America, as far as the Antarctica regions, and also it spreads across Europe and across Africa and Middle East and Asia. And this virus also causes a lot of human cases, especially in the United States, which is associated with the dairy cow cases. And this slide is also to show you the continuation of the outbreak, since emergence. As you can see, there were a lot of cases even in March 2025. That means, this virus undergoes, changes and so on. So we do expect maybe more outbreaks in the future based on, the seasonality of wild bird migration. And I really like, the topic of this meeting, because we have a shared risk, which is H5N1. That is our common enemy. And we need to find a shared solution, for this common risk. But are we really doing very well in finding a shared solutions? There is yes or no answers for that. But I think maybe, there are some issues related with research funding and issues with collaboration and competition, and also training. For example, this problem is also there in developing nations. So are we really doing very well in doing some training and also in resource sharing and so on? So the risk is there, it's really big, but we are not in a position to give a very soundful shared solutions for this problem. And coming back to the dairy, the ruminant livestock cases in USA and UK, this slide is to show you the primary source of infection is just wild birds, especially waterfowls or water-associated birds. These are the primary source of infection for ruminant livestock. As you are aware, there was a case of this virus, H5N1 infection in lactating goat in the United Kingdom, and also one case of B3.6 genotype in the USA, and the dairy herd is really hit harder with this outbreaks. At least there are 3 to 4 incursions from wild bird origin to the dairy cows in the USA, and this virus is spread across the multiple states in the United States of America. And we don't really, there is a primary source of infection. As I said earlier, these are wild birds. They are the primary source of infection, but we don't really know how this virus, invades, the mammary gland. If this virus was a purely respiratory disease in ruminant livestock, there is no surprise, because these animals, they inhale and the virus replicates in the respiratory tract, so and these animals showed, respiratory signs. But this is an unusual, for a virus to invade, the mammary gland. But there are a lot of hypothetical, some assumptions how this virus reached, the mammary gland. The first one was a mouse-to-teat transmission. For example, suckling calves, kids, or lambs, when they feed on pasture, which is contaminated with feces from infected, birds. So that means when these suckling animals, when they graze on this contaminated pasture, and at the same time when they suckle, that means they can readily transmit the virus from the pasture to the teat. And another one is in dairy cows, there is a condition which is known as caudal leaking. Sometimes the dairy cows, they clean, the skin folds between the udder and the inner thigh muscles. In that case, if the cows were feeding on pastures contaminated with this virus, at the same time when they leak, their udder area, they can transplant, the virus from the pasture to their teeth. And once the virus is introduced, replicates in the mammary gland, then what follows is a secondary spread. In that case, the assumption is a shared milking equipment, especially the shared milking cows. They are responsible. They are not the primary or the main driver. Other factors may be involved, but they are responsible. But recent studies even didn't support this hypothesis of shared milking cows, as the predisposing factor for the virus transmissions from cow to cow. And also recently in the USA, they found infectious virus in the reclaimed wastewater. That means, there is also a tendency for teat invasion from this wastewater. And another one, which we really undermine, is the respiratory exposure, because in the air samples, sometimes contains infectious virus. So the respiratory exposure, it also maintains, this infection within the dairy herders. So these are, the different mechanisms how this virus was introduced into this dairy cows and other ruminant livestock. And this slide is to show you the lactation curve in dairy cows. As you can see here, in the dairy cows, the lactation curve, it has early phase, it has a mid phase, and it has a late and the dry period. In one of the biggest farm in Ohio State, they studied, the prevalence of clinical influenza, and they found out that more clinical influenza cases involved cows in the late lactation than in the early lactation. For example, 40% in late lactation and 20% in the early lactation. Why such differences is there? We don't know. It may be related with the physiology of milking and some hormonal influence, immunological influence. There are other factors which can speculate. And before we did some animal studies, we studied, some of these receptors, the sialic acid receptors, the avian and the human type. They are responsible for the virus to enter into the susceptible host. As you can see in here, the mammary gland and the nasal tissues, lung from goat and sheep, they express, both the avian type and the bovine type receptors. That means in principle, this virus should replicate even in respiratory tract, not only in the mammary gland. They should replicate in the respiratory tract. But this is not the case. The virus has affinity to the mammary gland than the respiratory tract. Why it didn't replicate very well in the respiratory tract? We can give, some speculations and some hypotheses, but I think that we need to work further. And also we collected cells, the nasal cells, bronchial, small airway cells, and mammary cells. And we studied, the replication of a number of viruses in these cells originated from goat and sheep. Almost all viruses we tested, they replicate at high titers. The pandemic H1N1, it is not replicating in any of the cells. And then we did lactating goat experiment, and then we did a lactating sheep experiment. As I told you earlier, the cases in the dairy cows, they are more in the late stage lactation. So in our goat experiment, we used late stage lactating goats as a model. And we selected 2 viruses based on our in vitro studies. And we included the bovine or the cow H5N1 virus, the B313 genotype. And also we used the Canadian virus, which was B12 genotype, isolated from turkey vulture. And this virus has mammalian adaptive mutations at PB2701 position. And also we did some studies in ferrets, and this virus, it replicates, it's really transmitted readily between ferrets by contact. And also there was an aerosol transmission to contact ferrets. And we said maybe this is a very good candidate virus to compare with the cow virus. And as I told you earlier, in the dairy farm, the same cow may be infected by the respiratory route and at the same time the intramammary route. So the infection, we expect it is a combined routes of exposure, and we infected both udders in the lactating goats. And this is our experimental plan. We have 5 lactating goats, and we infected the same goat by intranasal, by nebulizer, aerosolization, and at the same time by the intramammary route. And then after 24 hours, we introduced suckling kits to see if there is transmissions from the mother, to the kids. And we collected, daily rectal temperatures, milk, yield, and swabs, and then blood samples. And we did also necropsy at 3 days and 6 days post-infection. And we collected milk. This shows, the milk samples which we collected, they showed altered, milk consistency and texture. As you can see, the milk there appeared, have a lot of clots, and also flakes. And also we just did, California mastitis test to show, the amount of, this somatic cells in the milk. As you can see, with the California mastitis test, it shows really, like gel formations and so on. As you can see, we saw this positive California mastitis test until the end of the experiment, until 40 days post-infection in all goats, lactating goats infected by both viruses. And also we recorded, the body temperature, the rectal temperature, as you can see in the upper panel. Both viruses, they cause pyrexia, not only in the lactating goats, but also in the kids. That means the kids are also infected with this virus. And also we measured the milk yield, the bottom panel, as you can see. There is a drastic reduction in milk yield in both viruses. And this really resembles, what we have seen in the dairy cows in the field, and also with experimental studies from dairy cows. And also we collected milk and we measured the amount of virus in the milk. As you can see in the left panel, that is a milk titer. At the first few days, you see large amount of virus in the milk, and then it declined over time. And the right panel is to show you we did a PCR on this milk sample. It is the same, the trend is the same. More virus at the beginning of infection. That means there is more viral RNA, and then it declined over time. So that means this virus is replicating in the mammary gland to higher titers. And this is the mammary gland to show some of the histology. In the control panel, you see this is non-infected lactating goat. As you can see, you see a very nice outlines of the alveolar space in the mammary gland, and you see the outlines. But if you compare that with the B313, that is a cow virus infected lactating goat, you see the mammary gland glandular structure. You see hypercellularity. That means there is a lot of infiltrations by mononuclear, polymorphonuclear cells. And also, if you see the IHC and the in situ hybridization, there is huge amount of virus replications in the mammary gland. Then that incites in inflammation. If you compare that with the B12, that is a Canadian virus which we used, that one also causes significant mammary mastitis, inflammation in the mammary gland, extensive infiltrations, by polymorphonuclear cells. And you see those dark spots, they are all over. So that means there is extensive virus replications in these tissues. Then this shows, that this virus, both viruses, they have the mammary tropism. And these goats, we infected them with a respiratory route. As you can see, we also saw some nominal less degree inflammatory lesion in the lung. The control panel, this is non-infected lactating goats by the respiratory route. As you can see, the air sacs, they're clear, nice, and there is no exudate, transudate in them. But if you compare that with the B313 and the B12 panel, you see a clear difference. So this virus, by the when it's applied by the respiratory route, it induces some minor inflammatory lesions. And also you can see there is a minor degree of replications in the respiratory tract. And we collected, also blood and milk, and also the BAL fluid, that is a bronchoalveolar lavage fluid. And we measured antibodies. As you can see, in these goats, in the serum, in the serum of both the lactating goats and in the kids, and also in the milk sample, and in the bronchoalveolar lavage fluid from the lactating goats and also from the kids, we were able to demonstrate, the significant inductions of antibodies. You see higher neutralizing antibodies. That means this virus is also eliciting some immunological responses. And once we have an idea about this virus in lactating goats, and that we showed, it replicates very well in the mammary gland. Then we studied in the sheep. And in the sheep experiment, we used early stage lactating, to see if, we see some mammary infections or not. And we also we first studied in the mammary epithelial cells of sheep, the replication patterns and kinetics of a number of virus. In this case, we used D11. The A6 genotype, it is an H5N5 virus. The rest, they are H5N1. We also included H5N5 just to see, this mammary tropism, is it exclusively, dedicated, to the H5N1 or any virus like H5N5 viruses can replicate in the mammary gland. So we also studied, the B313 virus. We compared its replication with the D11, and in vitro, we didn't see any difference. Then we said maybe we have to use the D11 genotype and the A6 genotype to see how they are infectious. And in this case, we had non-lactating sheep. We infected by the aerosol routes, and we have lactating sheep. We infected them by the intramammary route. In this case, we didn't combine the routes of infection, but we used, the intramammary routes and the aerosol routes for the non-lactating. And in this study, we infected the right mammary gland only because we knew that this virus replicates in the mammary gland of the goats and also in the mammary epithelial cells from the sheep. Then we infected the right mammary gland. The left mammary gland was not infected to see if there is transmission from the right mammary gland to the left by the suckling lambs. This is our experimental plan. The aerosol group, they're infected by aerosolization. The other group, they were infected by the intramammary route. In the intramammary routes group, after 24 hours, we introduced suckling lambs to see whether they get infected by suckling milk and whether they can serve as a vehicle of transmission of the virus from the right mammary gland to the left mammary gland. We did rectal temperature recording, swabs, blood sample, and necropsy at different time points. And this is for the non-lactating sheep, which were infected by the aerosol route. As you can see, when you infect them by the aerosol route, you see transient fever, one fever, and then it drops, and then they attain, the normal temperature. And we also did pathology on the lung tissues. And if you compare the D11 and the A6 is H5N5, D11 is H5N1 subtype. You see there is some degrees of inflammation in the lung. There is focal interstitial pneumonia, and also you see those brown spots. Those shows, the virus replications and antigen detection. And we collected from this group blood samples and bronchoalveolar laval fluid. As you can see in the serum, in both virus groups and in the bronchial alveolar lavage fluid, in both virus groups, you see antibodies in the serum and in the bronchial. So that means the respiratory route of infection, it induces systemic antibody responses, and at the same time, it may induce local immune responses. And coming to the lactating sheep, the results from the lactating sheep, as you can see, when we infect the lactating sheep by the intramammary route, they develop a fever. You see a number of spikes. That means not only in the lactating sheep, but also in the lambs. That means the lambs were infected. Lactating sheep was they were also infected. So that in both D11 and A6. And as you see the milk, you also see some degrees of changes in the organoleptic properties of the milk, the texture, consistency, and 2 of the sheep infected with the D11 virus, they have blood in their milk. That means maybe they have some secondary bacterial complication which exacerbated the lesions. The CMT score, the California mastitis test score, as you can see, there is a gel formation that is an indication that those milk samples from the mammary gland, from the right mammary gland, contains a lot of somatic cells because of the mastitis. And this slide is to show you the virus titers in the milk samples. In the right, the milk samples collected from the right mammary gland and the milk samples collected from the left mammary gland. This is a titer in the milk from the right mammary gland. As you can see, there are, higher titers, higher virus replication at the beginning, and then it declines. And as you can see in the left mammary gland, we see also viruses in the left mammary gland. We infected the right mammary gland. We also saw viruses in the left mammary gland. This shows that there is a transmission of the virus from the right mammary gland, which is infected, to the left mammary gland. As you can see, in one of the lactating sheep 2 to 4, we saw evidence of infections in the left mammary gland, so that we were able to collect, infectious virus at 5, 6, 7, 9 days post-infection. And that is in the D11 infected lactating sheep. In the A6, the H5N5, we saw the virus replicators in the right mammary gland. And also we saw there is a transmissions from the right mammary gland to the left mammary gland. That means this transmission pathway is definitely by the suckling lambs, because the suckling lambs, they suckle on the right, on the mammary, and they have unrestricted access to their mothers. That means they can transmit, the virus. So this shows there is a mouth to teeth transmission in this case. And also we do some sequencing on this, the lung tissues, which were collected from the aerosol infected non-lactating sheep. And as you can see, when the virus replicates in the various lung lobes, the virus, it shows, some, changes in nucleotides. There is some substitutions. But if you see the one of the goat, which lactating goat, where we saw transmission in the left, we sequenced, the viruses from the left, the milk samples collected from the left mammary gland. You see, compared with the inoculum, our inoculum virus, the D11 virus, it is a virus in transition. That means this virus at PB2, the basic polymerase 2 segment, it has a avian and mammalian mammalian signature amino acids. For example, at 627, this virus, it has E and K variants, and at 701, it has D and N variants. So that we selected this virus to see if during transmission, especially in the milk, to see it will be transitioned to a virus which is with a fully mammalian adaptive mutation. As you can see, the milk samples collected from the left mammary gland of one of the sheep, you see there is a significant changes. There is a number of nucleotide substitutions. And in this case, the virus, it fully adapted, it fully gained, the mammalian adaptive mutation at 701 from D, that is an avian type amino acid, to N, which is, the mammalian adaptive mutations. That means upon transmissions, these viruses, they can gain, this momentum. And this is a slides, the pathology and the IHC slides. In the left panel, that is the right mammary gland. As you can see, the D11 infected sheep, you see extensive hemorrhages in the mammary tissue, extensive virus replication, all those dark precipitators. And when we compare that with the A6, with H5N5, it is really, you see that there is a very clear difference. And if I take you to the right panel, that is the left teat cistern of the same ship, G168. As you can see in this ship, even in the teat cistern, you see virus replications, in the teat cistern. That means there is transmissions from the right mammary gland to the left. In this case, in this ship, because we euthanized by 3-DPI, and by 3-DPI, we saw viruses, replicating in the teat cistern. That means the transmission from the right to the mammary gland, it can occur, as early as 3 days in this particular ship, and then in another ship, from 5 days. So at least in the D11, we were able to see virus transmission from the right to the left mammary gland in 2 out of, the three ships. That is around, the 70% of, the cases. And this is, I brought this to show you this virus, when it infects the mammary gland, it also can infect, the draining lymph node, the supramammary lymph node. Each mammary gland has its own supramammary lymph node. As you can see, we can detect the viruses in those endothelial cells. The upper panel with the red arrows shows the virus antigens in the endothelial cells. That means this virus can invade, the vascular system. There is a tendency that this virus can enter into the vascular system and spread all over. And the bottom panel shows we detected, viral RNA in the uterus and ovary consistently in the lactating goat study and in the sheep. And this is to show you that this virus, even it can replicate in the uterus. That means the B313 and D11 virus, they have some effects on the reproductive performances of, these ruminant livestock. And this is also the supramammary lymph node. The upper panel is from the lactating sheep infected with the D11, and the bottom one is from the lactating goat, just to show you that these supramammary infections and detection of viral antigens, it can be for the D11 and B12 virus. As you can see, those dark precipitators, these are in the marginal reticular cells. This, we don't know why the roles of this marginal reticular cells. There are some few studies, but you can see these are networks of cells. Maybe they can more protective cells, and also they may play roles in antigen presentations and elicitation of this immunological response by transferring antigens to the follicular dendritic cells within this supramammary lymph node. And this is my final slide. And the upper panel is, the antibody titer in the serum of the lactating sheep and lambs. As you can see, there is large amount of neutralizing antibodies in the serum of the lactating sheep and in the serum of the lambs. That means, the lambs, they are infected. And the bottom panel is to show you the antibodies, the neutralizing antibody titers in the milk, in the lactating sheep. As you can see, we were able to detect higher titers of neutralizing antibodies in the milk samples collected from the right mammary gland, which was infected, and also in the left mammary gland. Even if there was no transmission, we were able to detect antibodies in the left mammary gland. That means, some of the systemic antibodies, they can be secreted into the mammary, into the milk. With that, I will conclude my presentation. Clade 2.344b, whether it's H5N1 or H5N5, they showed mammary tropism. These small ruminant models, they can be excellent to study H5N1 pathogenicity. And also if we study antibodies after vaccinations, and if we want to study how the longevity of these antibodies, their persistence, we can keep these animals in the containment, for some times. And the respiratory exposure, it can elicit strong antibody response, and we may use this route of infection if we have an attenuated H5N1 vaccines. And in this study, the suckling lambs, they can facilitate transmissions between teats from the right to the left mammary gland. And yeah, these are all, the guys who contributed into this study. And this is the end of my slides. And if you have any questions, I'm more than happy to answer. Okay, thank you.
Thanks, Tamaru. Excellent presentation. A lot of information, a lot of hard work crammed into 30 minutes. So thank you very much. It's very exciting. I see one question in the chat. One of the factors in increased virulence of high pathogenic avian influenza in late gestation could be lower mucosal immunity, which can facilitate virus replication in mammary gland. Maybe this is a comment. Do you want to comment on this one? So she's thinking that increased virulence is due to lower mucosal immunity. I don't know whether there are evidence to support that idea.
Yeah, it can be, I think that's, it can be one reason, so maybe there is a change in the profile of, the hormonal, from early to late, there is a complete shift of, this hormonal influence. I think we have to consider that into account. And also the milk, the amount of some factors, for example, if you take plasmine, which is responsible for cleaving the HA, maybe we don't know the amount of plasmine in early lactation milk and in late. That is also another one. And also we have to really dig into deeper to see, especially those gamma delta, the gamma delta T cells, they may be responsible, that these cells, they have both an adaptive and also that they have that innate like response, so that we have to look that into account. And also the amount of, the salicylic acid receptor, sometimes, they are not only attached to the cell surface, sometimes they can be, present as soluble form, We have to see, are there large amount of those soluble salicylic acid in the milk in the late lactation or in the early lactation. And also, we have to see the microbiome, that we can't say the mammary gland is a sterile environment. There may be some microbiomes, some bacteria may be there, so we have to study, these aspects as well. There is not one single predisposing factor. I think there are multiple, predisposing factors, they can be responsible for such variations in the, in the incidence of this, of the clinical cases.
Excellent. There's another, I think, comment in late lactation, immunosenescence of the immune cells may cause lower responsiveness to viral pathogens, changes in morphology of the mammary gland, epithelial cells with increased spaces between epithelial cells. So yeah, it looks like there are many factors that can influence, and so more studies has to be done to figure out what's going on. We have 3 more minutes. I think, Susana, you have a question?
Sí, gracias, doctor Alki. No sé si tiene activado su traductor, pero si no, puedo depositar la pregunta en el chat. Bueno, mi comentario era más relacionado al inicio de su presentación, donde efectivamente muestra el título de nuestro taller, que es Riesgos compartidos, soluciones compartidas, ¿no? Y efectivamente, como Procinorte, el objetivo es generar estos foros, ¿no? Para poder compartir información, buscar colaboraciones y encontrar soluciones, ¿no? Y en ese sentido, les comento que tenemos ahorita una convocatoria abierta sobre influenza aviar para que puedan proponer sus propuestas de investigación, efectivamente en colaboración con los investigadores que están aquí presentes o algunos otros panelistas, ¿no? O investigadores que ustedes conocen. Mi pregunta era más relacionada a los hallazgos que usted encontró sobre el tropismo del virus en la glándula mamaria. ¿Eso qué implicaciones tendrías para las medidas de control y vigilancia? Acabo de depositar la pregunta en inglés en el chat, por si quiere revisarla.
Tamaru, the question is, what implications do your findings on the virus tropism for the mammary gland have for disease surveillance and control?
Yeah, I think, we have to, especially in large dairy farms, I think there should be a routine, surveillance. I think that is done extensively in the United States of America, and also in Canada. We do a lot of this milk, bulk milk testing survey. And I think mostly we ignored, those farms with a mixed, livestock system. Some farms, they keep, the poultry, ducks, geese, together with, lamb, cows, and sheep. I think with that part we have to also take into consideration. So we have to target those as, also in our surveillance studies. So because as you can see, we do expect that this virus really infects the mammary gland, so that we have to really see how that looks like in other ruminant livestock. And also this, maybe I didn't present, we were in the oral swabs of one of the suckling lamb. We detected a virus which is already adapted, which was fully changed into the mammalian adaptive. That means this virus also replicates in the oral associated tissues. That means maybe handlers, those ones who handle this small calves and also lambs and so on. I think that we have to see now H5N1 is an agricultural problem, and from the extensive outbreaks in the USA, I think we have to see also dairy cows by the avian lens, so that we have to do good surveillance in that aspect.
Thanks, Tamaru. Yeah, I thank you, Tamaru, for your presentation. We have to move on to the next talk, and if there are additional questions in the chat, Tamaru, you can directly answer to the person who put the question in the chat. The next presentation is Dr. Belio Ruda, and the title remains secret, I guess. And I let to Bailey reveal the title. And you can thank you very much for willing to present at the meeting, and we are looking forward to your talk.
All right. Thank you all for the opportunity to present. I'm going to be discussing just a subset of the highly pathogenic avian influenza inoculation studies that we've been doing out at the NADC in pigs and cattle. So time is short. Let's move on quickly. Highly pathogenic avian influenza is a defined phenotype in poultry. These viruses have a run of polybasic amino acids in their fusion loop. This run of polybasic amino acids allow the virus to be cleaved by ubiquitous proteases. So no longer is this virus relegated to the upper respiratory tract and the GI tract of poultry species. It can now enter into, escape the endosome, enter into the cell, and start its replication outside of those sites. So it can go into the brain, into the heart, into the pancreas, and into the spleen and replicate. This systemic distribution causes high mortality. However, highly pathogenic avian influenza does not mean that it's going to have the same phenotype in other hosts. We don't necessarily understand why, and we cannot predict which virus or viruses pose the most risk to livestock based on sequencing. As we've heard previously, highly pathogenic avian influenza is an influenza A virus, so it has a segmented genome. As a result, it's able to do this really super nifty trick of reassortment and increase its fitness. So this occurs when two different viruses infect the same host, exchange gene segments, and produce a more fit virus out of that host. As we've heard, it's more of a them than an it. So in the United States, there are numerous different genotypes because of reassortment. So multiple introductions of highly pathogenic avian influenza have then reassorted with North American low-path avian influenza and resulted in these multiple genotypes. As you can see here, the more recent genotypes D1.1 and D1.2, they are only different by at 2% or less in their PA. So is this difference biologically significant to livestock? We don't necessarily know the answer to that. High-path avian influenza genotypes do wax and wane across different populations and time. As you can see from the beginning of the outbreak within the United States, it's affected here, wild birds, different mammal species. It's caused the loss of 174 million birds, as well as considerable amount of money in indemnity and compensation. It's infected livestock species, including cows, 1 pig, llama, and goats. We have also had multiple human infections associated with infected exposure to animals. Lucky for us, there are multiple adaptations that are required to overcome species barrier. So for instance, if I was infected with a high-path avian influenza, the first change that's likely to pop out is a change in the BB2 right here. And so that BB2 will change from an E627 to a K. So now it can play better with my host proteins and replicate more effectively within my cells. The next step would likely be a change from an alpha-2,3 to an alpha-2,6 binding predilection, followed by a balance between the HA and NA, so that the virus can swim through the host mucus and then also detach from the host cell effectively. So why do we do inoculation studies? Well, we want to assess which viruses pose the most risk and what do they look like in pigs and cattle. Importantly, these studies do generate known status samples. These are necessary or paramount for assay development as well as assay evaluation to get new tests and the best tests by sample type. Best sample types for detection and monitoring may also be able to be identified. We understand the time course of disease as well as the immune response. It develops models for vaccine evaluation, and the results that have been generated may be used to inform response planning. There are limitations to inoculation studies. A positive is important, a negative not so much. Why is that? Well, animal numbers. We are limited on the number of animals we can put into our BSL-3 egg facility. If you're looking at a typical barn within the United States, it has 1,000+ pigs. We're looking with maybe 20 pigs per group in our studies. Our BSL-3 egg in a room, it exchanges the air 6 times within an hour. That is at no way comparable to what happens in a barn. These animals are under limited stressors and they are not infected with other primary pathogens. So why are we worried about swine? Well, there have been periodic detections or incursions of low-path avian influenza into commercial swine, both in Canada as well as the United States. The continued circulation across different animal populations puts swine at higher risk. Highly pathogenic avian influenzas in mammal species are known to have a systemic distribution, and they can traffic via nerve axons to other locations, which could pose a public health risk. There is potential for mammalian adaptation and or reassortment resulting in circulation in swine. Endemic influenza in swine is very common in commercial production. There is a lot of genetic diversity within the U.S. herd, and these clades, just like the high-path clades, do wax and wane within our herd. These viruses contain human-adapted gene segments, and so again, reassortment is a concern. What have we found in swine so far? So we have found that different high-path influenza virus strains pose different risks to swine. They are able to replicate within and outside of the respiratory tract of pigs, and macroscopic lung lesions consistent with influenza may not necessarily be present in an infected pig. 3 out of the 4 high-path strains that we've used that have the E627K BB2 mutation, which is thought to be a mammalian adaptation marker, did transmit in our model. Based upon the strains that have been detected within the United States, about 20% of the strains from mammalian hosts do have this marker. One of the strains that we have used in our swine model with a D701N BB2 mutation did result in the most severe clinical disease in primary inoculated pigs. However, this mutation is rarely detected in strains from mammalian hosts. This is our typical study design. So we're using a MAD device to inoculate intranasally 1 ml. So we are not doing intratracheal inoculation, and we are not giving them a bunch of virus within their nasal cavity. It's a pretty minimal dose that we're giving these guys. We're then placing contact pigs at 2 DPI to look for transmission. We will take some of the primary inoculated pigs at 5 DPI to look at virus distribution, where has the virus gone within those pigs. And the remaining pigs will be left from anywhere from 24 to 35 days post-inoculation to monitor for seroconversion. We have taken different sample types throughout the studies, including oral fluids. However, we can detect the virus within oral fluids at 100% prevalence. We do not design our studies to understand if oral fluids would be a good sample type for surveillance. This is a summary of what we found by strain so far using our model. Because it's a summary slide, there are going to be some nuances that have been lost. So in essence, all of these viruses do at some level pose different risks to swine. The strain ID is on the left, followed by the genotype and whether or not that strain had known adaptation markers. All of these viruses could replicate in the lower respiratory tract of pigs. However, the number of pigs in which it was detected and or the viral titer at which it was detected can be different. We were able to detect it consistently in nasal swabs in a majority of these viruses. However, again, it's going to vary. So the ones that are in red are the strains in which the amount of virus being detected in the nasal swab would be similar to what you would expect from an endemic influenza. We also can detect it in fecal swabs. The turkey vulture California B3.2 was actually detected more in fecal swabs than it was in nasal detection, so that is why that has a red yes there. We can detect transmission in, again, a subset of these viruses in our BSL-3 egg. Respiratory signs when seen with these viruses are commonly mild. We have seen neurologic disease using a turkey vulture B3.2 D701N in 4 of the primary inoculated pigs. Neurologic disease consisted of we saw circling, stargazing, as well as seizures, and that occurred 7 days after inoculation. Gross lung lesions, when present, look like your typical influenza, and we are also able to detect it outside of the respiratory tract in a subset of the pigs, including the brain, pancreas, heart, meat juice, and diaphragm. So looking at the path, we can detect it within the endothelial cells in the lung, the pneumocytes in the conducting airways. And because these alveolar septa are wide, it is also causing an alveolitis, which isn't something that you would typically see with a host-adapted influenza. Here's the olfactory epithelium of the ethmoid, and you can see that's infecting cells within the epithelial layer as well as the subjacent axons. So it is trafficking from this epithelial layer into these axons. This is the brain of a pig, and obviously that you can see that it's within the neurons of the brain within the laminar area of the cerebrum. This is the pancreas, and then a duct within the pancreas, so it's able to replicate within the epithelial cells of the ducts of the pancreas in pigs. Looking a little bit closer at a more recently characterized virus, so we did a D1.2 P-fowl virus. In primary inoculated pigs, we could detect it in all of the pigs inoculated at 1 and 2 days, anywhere from pretty low detection to 6 logs of virus. At 5 dpi, we could detect it within the olfactory bulb, as well as the Peyer's patch of the ileum, within the diaphragm here. And then if you look a little closer at that area of staining, you can see that it trafficked to that location in the diaphragm, probably by that adjacent axon, which is also IHC positive. Typically, we do not take histologic lesions at 35 dpi. But because it was only 3 pigs and a student and I, we decided to take all of the tissues at necropsy. And lo and behold, in one of those 3 pigs, we were able to detect the virus by PCR, see consistent lesions within the CNS of a viral etiology. So here's a CD3 stain, and you can see that there's CD3 positive cells within the CNS. And then if you look at this, you can see that there's actually still a virus that can be detected by IHC within neurons and other cells within the CNS that correspond with the lesions present there. We could also still detect the virus within the epithelial layer of the ethmoid at 35 dpi. So what have we learned so far with using our model? Pigs are at risk for the North American H5 circulating strains. Typically, we see subclinical or mild respiratory disease. Occasionally, you can see CNS disease. We are starting to see more vomiting and diarrhea in a subset of those pigs about 7 days post-inoculation. So because they can be infected with these viruses, reassortment with endemic swine strains is a concern. Best sample type, historically, if you ask me what the best sample type was, I would be hard pressed to tell you because nasal swabs didn't seem to be so great. In more recent studies, it does look like nasal swabs are a pretty good sample type. It does depend, so base it on your clinical signs or the lesions you're seeing. If you're seeing neurologic disease, you think it might be high path, well, go after the brain first. Anything else is supporting. 3 pigs might not necessarily get you your answer unless you're really great at defining a case definition and then making sure that those pigs that you've just identified that meet your case definition and getting the samples from those pigs. So risk of incursion is likely low. Avoid untreated surface water. Ensure bird proofing. Restrict scavenger mammals. Don't feed raw milk to swine. And then evaluate the biosecurity risk posed by other livestock species, including the use of the labor force and resources such as trucks. So if H5 enters commercial swine today within the United States, we do have a lot of strengths. We have a pretty good surveillance system and diagnostic expertise. We're working on a response planning. We have relatively good biosecurity in our breeding herds. We may have some cross protection due to endemic swine. We have influenza vaccines, not H5 vaccines, but endemic influenza vaccines that have shown performance against endemic strains. And we have characterized multiple genotypes and adaptation markers in the weaned pig model. However, vulnerabilities do remain. Producers are hesitant to submit to the surveillance system. The response plans are still in draft form. They are suggestions, and it is difficult to construct these response plans with so many unknowns. In the United States, we have a continuous need to move pigs. The cross protection is currently unknown with variable population immunity. H5 vaccines are not licensed and are not allowed under autogenous or prescription. We will have further complications compared to cattle because we have so much endemic influenza circulating that we will not be able to use NP ELISAs for a DIVA strategy. Many of the strains that we've looked at have had variable clinical presentations or have had subclinical disease. Prevalent genotypes do continually change, and we do not know the risk to the lactating sow. Moving on to some of our cattle studies. So the study A has been published in which we found fibrosing mastitis, bronchiolitis obliterans, as well as antibody response and viral shedding. Study B is a dose response. So what dose can mammary gland be infected with, as well as pathogenesis. And then study C is the protection of convalescent cows. So study A, the study design looked like this. We took 2 lactating dairy cattle and intramammary inoculated contralateral quarters with 10 to the 5th 1 milliliter and took samples over time and euthanized those girls at 24 DPI. We saw an incredible amount of virus coming out of that mammary gland. So the mammary gland is essentially an awesome viral factory. And then we could no longer isolate virus where that box is located here. We were able to detect diagnostic antibodies as soon as 7 DPI, and then saw neutralizing antibodies in the milk around 9 to 12 DPI in inoculated quarters, with a slight drag in a non-inoculated quarters. In the bottom is a non-inoculated quarter, so that you can see that the majority of the section should be white. And so this is your secretory alveoli. Here is some blue. This is your normal supporting connective tissue that's found in the mammary gland. And then here you're seeing too much pink and not enough white. And so this blue is a trichrome, it's a fibrosis. And so you're getting too much blue in between these secretory alveoli. And this could lead to future production losses. It was a little crazy to me when I looked at these slides, but you can see influenza antigen all the way out to 24 DPI, at least when I was taught. Getting detection by IHC of influenza out to 10 DPI was a stretch, so 24 certainly is an additional stretch. And not only can we detect antigen at 24 DPI, we can also detect antigen within the nucleus as of a subset of these cells, which would be consistent with the ability of this virus to continue to replicate within the mammary gland at some level. In this study, we also nebulized 4 heifers and took 2 out at 7 DPI and 2 more at 20 DPI, taking all of these different sample types. We were able to isolate from the nasal swab of 2 of these girls. And we were able to see lesions within the lung. These lesions were super tiny, so you needed good eyes and bright lights. However, the lesions were tiny but real. And so what you're looking at here is a conducting airway. This is the epithelium lining the conducting airway, and here you have an inflammatory infiltrate. And so gold standard practice to say that these lesions were caused by this virus would be able to detect the virus using an in situ assay. So here you have influenza antigen detected within the nucleus of the conducting airway epithelium, demonstrating that this virus led to this lesion. In these bottom panels, you have bronchiolitis obliterans, and again, very faint detection of influenza antigen within this affected airway. I'm going to say that if you really wanted to see optimize the detection of influenza within respiratory lesions, at least using this model here, I would probably take these girls out at 2 or 3 DPI rather than waiting till 7, but we were also trying to assess seroconversion. So we got lucky in that we were able to detect seroconversion in these girls, as well as antigen and lesions within their respiratory tract. Virus detection by qPCR. In the calf, we could detect it in the lung, lymph node, as well as turbinate. And in the cow, we were able to detect it in the inoculated mammary gland, as well as lymph node. Study B is kind of a study within a study within a study. And so this study provided infected milk for transmission to calves, milking equipment transmission, as well as oronasal exposure of cows. So we had 6 different cows. We used 3 different inoculation doses, 1 per quarter, and left 1 quarter uninoculated. We then took cows out at different days post-inoculation. Similar to the first study, we did see changes in the milk from 3 DPI across all of these different inoculation doses. So in the bottom, you can see 10 to the 2nd, to the 4th, and 10 to the 6th. And then the California mastitis test, we also looked at color as well as consistency. Again, we saw production losses across the cows that were inoculated, as well as virus detection within all 3 of the inoculated quarters. When looking at these quarters grossly, and if you have a good knife, in a non-inoculated quarter, the quarter should really cut like butter, it should leak milk, and it should look like a cauliflower. If you're cutting through an infected quarter, it'll cut a lot more, the cutting will be a lot more challenging, and the inoculated quarter will look more like a pork chop and also feel more like a pork chop as a result of the fibrosis. As you can see in the bottom image, there is pathologic atrophy, and that pathologic atrophy of the gland corresponds to the amount of virus that was given within the gland. So 10 to the 2, it's smaller than a non-inoculated quarter. However, it's still bigger than 10 to the 4th. And then the smallest quarter is 10 to the 6th. Microscopic evaluation, so RNA scope and antigen detection at 3 dpi. We could also find it in the lymph node as well as a cardiac myocyte by IHC at acute time points. The middle panel is vessels within the mammary gland, so you can find it by replicating. This target is M replicating probe, so you can detect M replicating RNA within this vessel. You can also detect it by IHC in different cell types within the vessel, as well as endothelial cells within a vessel of a mammary gland. These are more chronic stages of disease, and so what we start seeing is these antigen aggregates. And so you can see these antigen aggregates with an intense inflammatory infiltrate surrounding those. At this space and time, you can also see antigen still present at 7 and 28 dpi, and there is a cell there that you can see that it's still replicating within the nucleus. Study C is protection from convalescent cows. What we did is take infected milk and pour it in their feed bunk over an 11-day period. After 20 days post-exposure, we then inoculated the quarters with different viral titers and took them out to necropsy at 7 days. We did not see a significant impact on milk production. They did seem to shed the virus based upon PCR, however, not to the extent that we would expect if they were not protected. So going back to the first study, this is where we would see PCR ranges for this duration of time. And here we have definitely higher CT values in these girls that had neutralizing antibodies. So cattle applications. We've seen mammary fibrosis, inflammation, and persistent antigen that may lead to decreased milk production and future lactations. The severity of diminished production will likely correlate with the number of affected glands and the infection dose. Viruses can be detected outside the mammary gland at acute time points following inoculations, and heifers could be a source of virus transmission through respiratory secretions. Convalescent cows with systemic antibodies were protected from clinical disease. However, this is going to likely be, the amount of protection will likely be reflected upon the recency of exposure and the amount of antibodies they generated, as well as the dose in which they are exposed. The complexity of influenza ecology and impact appears only to be increasing. The risk of high-path avian influenza becoming endemic in cattle. There's also a risk for high-path incursion into swine. This really speaks to the importance of cross-sector collaboration. Thank you all. All of these studies definitely take a village to conduct. Are there any questions?
Thanks, Dr. Aruda. Excellent presentation. A lot of information. Let me see whether there are questions. Yes. Now there's a question from David. He says, I might have missed this, but was the localization of virus in pigs in ethomodial axons and brain associated with any pathology?
Yes, so there are lesions within the brain. So for instance, in this most severely affected section, it actually looked like laminar cortical necrosis if you looked very closely, but there's also inflammation within the meninges and lesions consistent with a viral etiology there. So yes, there are lesions. You can see lesions within those brains.
Okay. And I think here's another question. Were peripheral axons affected anywhere else in the body?
Good question. Don't know for sure.
Okay. Was there any associated neuropathology? If not, what do you make of that? Maybe related to this question, were the tissues slides in IHC or ISH? Did you do qPCR on brain?
Yes, we did qPCR on brain. Some of the CT values ranged from 12 to 16 in the affected pigs. In pigs that we cannot appreciate CNS disease. At the time of necropsy, we can actually still detect it sometimes at 17 to 19 CT PCRs. So it's pretty amazing that it can be there at that level, and we still can't appreciate clinically that these guys have a virus in their brain.
That's very interesting and scary at the same time. So
Yes, they must have more brain than they need.
Thank you very much. I think we are running out of time, so we will move to the next. Thank you, Dr. Arruda, for the nice presentation, and we'll move to the next talk. This is from Mexico, National Autonomous University of Mexico. Sorry, I think I'm messing up with this. So National Institute of Respiratory Diseases in Mexico. Dr. Perez will talk about where we stand on avian influenza in humans in Mexico. Dr. Perez.
Hola, buen día. ¿Pueden ver mis diapositivas?
Sí se ven.
Muy bien. Y voy a apagar un momento la cámara para que no se interrumpa la presentación.
Dejamos de ver su presentación, doctor Joel. Listo, gracias.
Buen día y gracias por la invitación. Cambiamos de tema, ¿no? Un poco, porque lo que hacemos nosotros es la caracterización de influenza en humanos. Y el objetivo de esta plática es charlar, mencionarles los hallazgos que hemos detectado caracterizando los virus de influenza aviar en humanos en México. Primero vamos a mencionar algunos datos importantes de la biología del virus, será breve ya que ya se ha tocado el tema, y posteriormente un resumen de lo que hemos encontrado con el virus de influenza en humanos aquí en México, en específico en el INER. Y por último, les presentaré los datos que hemos encontrado detectando los casos en humanos con influenza aviar. Entonces, solamente para, recordemos la estructura, ¿no? del virus de influenza. Es un virus que pertenece a la familia Automyxoviridae. Tiene una característica muy especial, que es un virus con RNA segmentado de polaridad negativa, y que las proteínas de envoltura neuraminidasa y hemaglutinina son las que van a determinar el subtipo del virus. Importante señalar que tenemos como muy determinados cuáles subtipos causan enfermedad común en humanos. Son los virus de H1N1 y H3N2. Anteriormente se había detectado H2N2, ya no más. Hace mucho tiempo no se ha detectado. También el virus de influenza B. Sabemos que hay dos clases, Victoria y Yamagata, del cual ya solamente está actualmente el virus Victoria. Y de forma esporádica, el virus de influenza aviar, H5N1, H5N2, H7N3, H9N2 y algunos más, causan infecciones tanto respiratorias como moderadas, principalmente conjuntivitis en humanos. Y cuando pasa esto, ¿no? Los virus de influenza aviar pueden ser potencialmente pandémicos. ¿A qué se debe este cambio del virus de influenza? Pues a su propia característica biológica. Su RNA polimerasa se equivoca mucho y incluye en cada ciclo de replicación diferentes mutaciones. Esto vamos a llamarle genetic drift. Y también hay otro mecanismo en el cual ya se ha comentado, el rearreglo de segmentos entre diferentes subtipos puede dar como lugar a un nuevo subtipo. De tal forma que los diferentes subtipos de influenza que se encuentran en aves, y en humanos pueden eh, hacer que si se infecta un hospedero como es el cerdo, que tiene ambos ácidos siálicos, alfa 2,3 y alfa 2,6, dentro de este hospedero puede hacerse los arreglos genéticos de los virus de influenza aviar con los de influenza humano y entonces el cerdo es el principal hospedero para que se lleve a cabo este procedimiento de rearreglo. Este es un resumen de los casos que ha habido de influenza aviar en humanos. Vemos que desde los 90 ya empezamos a ver algunos casos con H5N1, con H9N2, con H7N3. De hecho, uno de los casos que voy a presentar aquí es en México en 2002. Posteriormente se han dado casos con H7N7. Y en los últimos años se ha detectado infecciones con H7N9 que tiene una alta tasa de mortalidad y detectamos el otro caso que les voy a presentar de H5N2, el primer caso en humanos y que fue fatal. Un momento, ahí está. Em, em, Existen subtipos de influenza aviar y hay una transmisión entre especies. Sabemos que el principal reservorio es en aves, es el primer reservorio de influenza tipo A y que está de una forma en homeostasis. Muchas veces son asintomáticas esas infecciones y que las aves son permisibles a todos los subtipos del virus de influenza. De tal forma que el virus de influenza en aves es una zoonosis continuamente reemergente. Y quiero tocar tres puntos importantes en cuestión de cuáles son los requisitos que debe de tener el virus de influenza aviar para que pueda causar una nueva pandemia. Es la transmisión de un nuevo subtipo, como ya lo vimos, principalmente por los arreglos de los diferentes subtipos. Posteriormente, la capacidad replicativa, que tenga una alta capacidad replicativa y que pueda causar enfermedad. Y por último, la eficiente transmisión humano a humano. De tal forma que entonces necesitamos buscar en todos estos subtipos de virus de influenza que causan enfermedad en humano, el patrón de rearreglo que pueden estar sufriendo y los cambios que puedan estar determinando adaptación a mamíferos, como son mejor afinidad receptor alfa 2,6 que tiene el humano, mutaciones en ese 1 de adaptación para evitar la respuesta inmune innata y también mutaciones en las polimerasas que van a permitir mayor actividad de la polimerasa y mayor replicación en células humanas. De tal forma que, concluyendo esta parte, el virus de influenza evoluciona por dos tipos de mecanismos, ya lo sabemos, por la deriva génica y por el rearreglo de diferentes virus en un hospedero determinado. Este rearreglo de segmentos puede dar lugar a nuevos subtipos que potencialmente infectan humanos, pueden causar pandemia y que la continua interacción entre los virus aviares y mamíferos, principalmente en cerdos, incrementa la emergencia de una nueva pandemia debido al virus de influenza. El primer punto es entonces mencionarles lo que estamos haciendo en México, en el INER, caracterizando el virus de influenza humano. Lo hemos hecho con el virus H1N1 desde su surgimiento en 2009 hasta 2019 y ya lo reanudamos también hasta 2025. Les muestro aquí una línea del tiempo de donde hemos caracterizado en México el virus H1N1 y hemos notablemente detectado algunas mutaciones de patogenicidad, como es en la posición 222 de la hemaglutinina y que confiere mayor afinidad de vías respiratorias altas a vías respiratorias bajas y lo hemos asociado con mayor severidad de la enfermedad y con mayor mortalidad. También lo hicimos en un modelo animal con la colaboración de la gente del Laboratorio Nacional de Microbiología allá en Canadá con el doctor Darwin Cobasa. Y lo hemos hecho a través del tiempo para detectar también mutaciones que podrían estar implicadas en la evasión de la respuesta inmune. Pausamos un momento después de cuando apareció el SARS-CoV-2 en 2019. Y les muestro aquí en esta diapositiva las sustituciones que han sido localizadas, ¿no?, en los principales sitios inmunogénicos de la hemaglutinina en H1N1 desde su inicio en 2009-2010. y a través del tiempo hasta la fecha de 2025. Hemos visto ya en los últimos años una gran inserción de mutaciones, cambios en las posiciones más importantes de los sitios inmunogénicos. Últimamente ya estamos también haciendo o concluyendo la parte de filogenia, tanto de H1N1, tanto como H1N1 y también H3N2, y estamos detectando las mutaciones más recientes y los linajes más recientes de ambos subtipos. Esto porque tenemos una colaboración con una red internacional de vigilancia del virus de influenza y enfocadas específicamente a virus de influenza en pacientes hospitalizados, de tal forma que estamos buscando estos determinantes de patogenicidad y asociarlos con la parte clínica. De igual forma, estamos haciendo también la filogenia del virus H3N2 en México. Tenemos menos historia de este subtipo de virus. Apenas en los últimos años comenzamos a secuenciar. Estos árboles filogenéticos que les muestro son con las secuencias de aquí de México y tenemos que construir la historia de H3N2. En México no se había secuenciado de tal forma que no tenemos tanta antecedente, y también lo estamos haciendo con esta red global de influenza para detectar el virus de influenza en pacientes hospitalizados. ¿Y qué ha pasado con lo que hemos detectado? Bueno, les quería mostrar lo anterior porque, dado que tenemos este proyecto de vigilancia genómica en el INEL, en este hospital, y tenemos esta colaboración a nivel internacional, hemos podido, ¿no?, detectar los casos más graves que han ocurrido en México con el virus de influenza aviar. De tal forma que les muestro los tres grandes brotes que hemos tenido en México desde el 2002 con H7N3, con H5N2 y con H5N1. Les muestro el primer caso, es H7N3. Les muestro los datos que se han reportado en la Organización Mundial de Sanidad Animal, y tenemos que desde el 2012 tenemos el primer brote en Jalisco, que es la parte central de México. En aves domésticas se detectó este brote en 2012. Si se fijan, tenemos prevalencia de H7N3, perdón, desde 2012 y hasta este año que seguimos teniendo casos de H7N3. De tal forma que aún no ha concluido la erradicación de este virus en México. La mayoría de los brotes han sido en los estados como Jalisco, Puebla, tanto en granjas de aves como en aves de traspatio, en los estados del centro de México principalmente, y algunos también en el norte del país como Coahuila y Durango. Y ahí, desde 2012, el primer caso de infección en humanos por H7N3, de este virus aviar, lo detectamos en dos casos en humanos, personas que trabajaban en granjas en Jalisco, y tuvimos dos casos con conjuntivitis. No tuvieron una infección respiratoria, no cruzaron con fiebre, y se asociaron con H7N3, se detectó en conjuntiva por medio de PCR tiempo real, y esta detección se hizo en humanos. Fue el primer caso. La PCR se hizo con un lavado ocular, y lavado ocular se utilizó para pasarlo a embrión de pollo y para posteriormente hacer secuenciación completa. Se publicó este dato en ese año, en 2012-2013. Y lo que encontramos es que este H7N3 es aviar, es de alta patogenicidad. Al ver los sitios de corte de la hemaglutinina, vemos que tenemos un incremento de aminoácidos básicos en el sitio de corte, de tal forma que pudimos determinar que este virus H7N3 que causó en ese momento el primer brote en México documentado es de alta patogenicidad. Cuando hicimos el árbol filogenético, el estudio filogenético, encontramos que efectivamente el H7N3 encontrado en humano estaba totalmente relacionado con los virus de granjas del brote de Jalisco del 2012, como lo mostramos en este árbol filogenético utilizando hemaglutinina. Y para saber si de dónde estaba estaba asociado este brote de H7N3 en México, hicimos un análisis filogenético para ver la relación con otras virus de H7N3 en Norteamérica. Vimos que había una estrecha relación con aves en Norteamérica, principalmente en Estados Unidos y Canadá. El siguiente caso que les voy a mostrar es el de H5N2. El virus H5N2 en México, la primera evidencia de este virus aviar fue demostrado serológicamente en 1993 y a partir de ahí fue aislado. En 94 se detectó otro brote de virus de baja patogenicidad, fue aislado de aves domésticas y en el 95 se detectó por primera vez un virus de alta patogenicidad, esto es también en el centro del país. Em, a pesar de que hay grandes esfuerzos de vacunación desde 1995, el virus de baja patogenicidad H5N2 ha permanecido exótico en la población de granjas en México y de nueva cuenta un H5N2 de alta patogenicidad ha sido detectado en el 94 y posteriormente en el 2024. Generalmente lo que se ha encontrado a nivel molecular es que el sitio de corte de hemaglutinina de los diferentes aislados desde 2016 al 2019 tienen o muestran un patrón que parecería ser asociado con virus de baja patogenicidad, que se tienen solamente dos aminoácidos básicos o cuatro. ¿Qué pasa entonces con los datos de H5N2, los brotes en México? Solamente hemos detectado o se han reportado ante la Organización Mundial de Sanidad Animal dos brotes, el 2015 y el de 2024. El primer brote fue en el norte del país, en el estado de Sinaloa, y el último que hemos detectado, que se detectó, fue este brote en Michoacán. Les señalo este último brote porque a partir de ahí fue el hallazgo del primer caso en humanos de este virus aviar. Un trabajo muy interesante ya anterior a esta detección se hizo con otra instancia en México, Senacica, secuenciando los virus H5N2 desde antes de 2022 y haciendo este estudio del ancestro común más reciente. Se estimó que el virus se originó desde 1990. De hecho, el dato coincide analizando cualquiera de estos segmentos y lo que se ve es que hay un solo origen de estos H5N2 y a partir de ahí, de hecho, se da origen a otros virus de Centroamérica como El Salvador y Guatemala. Y también algo interesante es que este H5N2 en México formó al menos 5 diferentes clados, hubo diversificación en 5 diferentes clados a partir del 2019. Eh, ¿Por qué les muestro esto? Es porque es el antecedente directo de lo que encontramos en el primer caso de influenza aviar H5N2 en México. Lo detectamos en un paciente hospitalizado en esta institución. Llegó del Estado de México, es la parte central, muy cercana a la Ciudad de México, y llegó con infección respiratoria aguda grave. Este paciente fue admitido a finales de abril de 2024. Cabe señalar que este paciente tenía comorbilidades, tenía daño renal y tenía ya esta afección durante muchos meses. El diagnóstico de inicio en el paciente fue que fue influenza positivo, pero no fue positivo, no fue negativo, perdón, para el virus H1N1, para H3N2 y para influenza B. De tal forma que pudimos realizar el ensayo de amplificación de genoma completo de influenza y pudimos tener 5 segmentos completos de NS, M, NA, NP y hemaglutinina. y de forma parcial PB1, algunos reads de PB2 y PB1 no tuvimos ninguna secuencia. Sin embargo, con estos datos hicimos el primer ensayo de BLAS para conocer la identidad de las secuencias encontradas. Y lo que encontramos de forma sorprendente es que este virus tenía una alta homología, 97-98%, con un virus de Querétaro del 2019. Y cada uno de esos segmentos, de los 5 segmentos que pudimos amplificar, tenía esta homología, pero trabajando de cerca con la otra instancia mexicana como Senacica, pudimos hacer un análisis mucho más completo. De tal forma que el virus de H5N2 estaba muy relacionado con este brote de en Texcoco en 2019. Encontramos que hay solamente 3 mutaciones que difieren entre este virus de 2019 y de 2024 del INER, en estas posiciones, solamente en hemaglutinina, en NS1 y NP. Eh, mm, no encontramos en hemaglutinina algunos marcadores que podrían estar asociados a adaptación, y dado que no amplificamos completamente el segmento PB2, no pudimos detectar si había algunas mutaciones asociadas a adaptabilidad. Sin embargo, vimos que en hemaglutinina, el sitio de corte de H5N2 tenía 4 sitios básicos en este lugar. Por estudios previos, la gente de aquí de Senacica había asociado ya con estudios funcionales que estos 4 sitios se estaban relacionados con H5N2 de baja patogenicidad. Ya con los datos de secuencias de H5N2 desde 2022 de aves aquí en México, en diferentes estados de la República, pudimos construir un estudio de filogenia, un análisis filogenético, para ver de dónde estaba asociado o cómo dónde surgía nuestro virus H5N2. Lo que encontramos es que había el virus de 2019 en Querétaro dio lugar a los virus de 2024 en México, y a partir de esto hubo la infección, o bueno, más bien la relación del H5N2 en humanos en México. Y así de esta forma pudimos relacionar que este virus 2024 se parecía al virus aviar del centro de México. Y por último, el caso de H5N1 en México, sabemos que es de gran atención el virus aviar H5N1, porque estamos viendo que puede ser una nueva amenaza, y puede ser el causante de una nueva pandemia. De tal forma que ya lo hemos visto anteriormente en las 3 presentaciones anteriores, que el H5N1 ha aparecido desde los años 90, ha empezado a causar también alta mortalidad, y que a partir de 2014 se ha dispersado en hasta 60 países en casi todos los continentes. Y este virus H5N1 se ha diversificado en diferentes clados y subclados, y que el dato hasta ahorita, hasta 2025, en julio, se han detectado a nivel mundial 985 casos en humanos y 473 muertes, de tal forma que hay una tasa de mortalidad del 48%. Y algo importante es que también el H5N1 ha reemergido, y se ha arreglado para formar nuevos subtipos como H5N6, H5N8, H5N5 y H5N10. Sabemos que a partir del año 2021-2022 hay una gran cantidad de brotes de virus aviares, y principalmente por H5N1. También ya nos han comentado cuál es la historia reciente del clado 2.3.4.4b, que se dispersó desde Norteamérica, Canadá, hacia Estados Unidos y posteriormente a México y Centroamérica. De tal forma que hay una gran cantidad de animales infectados, de tanto aves como mamíferos. Aquí está una actualización de los brotes actualmente, desde 2023 hasta 2025, y vemos la cantidad de países afectados en estos años y el número de granjas que han sido afectados en estos países. Y estos datos son reportados a la Organización Mundial de Salud Animal. Y sabemos también, ya lo han mencionado, la emergencia de este genotipo de 1.1, y que este clado 2.1 pues tiene la peculiaridad de ser constantemente encontrado en infección en mamíferos y se ha detectado en brotes en más de 30 especies de estos mamíferos. Estamos buscando entonces mutaciones de adaptación de aves a humanos, como estas mutaciones en PB2 o en PB1, y también mutaciones que pueden estar aumentando la afinidad de hemaglutinina para el receptor alfa-2,6. Como les mencioné, hasta julio de 2025 tenemos 985 casos en humanos y estos están asociados con los genotipos B3.13 y D.1, tanto en humanos como en ganado vacuno y en aves domésticas y de granja. El caso de México, primero les doy los antecedentes de los brotes en México de H5N1 causados por H5N1 y vemos que se ha detectado de alta patogenicidad desde 2022 en el Estado de México, que es un estado contiguo a la Ciudad de México, en estados también, tanto del centro como del sur, se ha encontrado también detección de H5N1. Y en 2023 también en el centro del país y en el sur del país, en el norte también del país se han detectado H5N1 de alta patogenicidad. Y por último, el brote que hubo en 2024 que no se ha resuelto en Durango, en el estado del norte del país, en aves silvestres y aves de granja. De tal forma que el primer caso de H5N1 en aves que se detectó en México fue un niño que tenía, de hecho es una niña que tenía 10 años en el estado de Durango, donde ya les mostré que se dio el último brote y que los síntomas empezaron el 7 de marzo del 2025 y el 13 fue admitido al hospital debido a falla respiratoria. El caso fue transferido a un hospital de tercer nivel y falleció desgraciadamente el 8 de abril por complicaciones respiratorias. Cuando se hizo la detección molecular, se pudo detectar que era del subtipo H5N1. Posteriormente, el INDRE, que es la institución de diagnóstico y referencia del país, hizo secuenciación de algunos segmentos y parcialmente de neuraminidasa, completos NS1, M, NP y hemaglutinina, y pudo caracterizar que el virus fue de este clado y del genotipo de 1.1. Yo hice esta árbol filogenético, todavía no se reporta el caso, y sin embargo ya están las secuencias en GSAID, podemos hacer este análisis, y lo que hice es hacer este análisis filogenético con aproximadamente 1800 secuencias de 2025 de aves mamíferos de Norteamérica, y lo que pudimos ver que efectivamente el virus H5N1 de México se encuentra dentro del clado de 1.1 y sorprendentemente se parece a H5N1 de aves de Virginia y Nebraska de este mismo año. Hay dos mutaciones interesantes en la posición de hemaglutinina. Tenemos que trabajar más acerca de la relación que tiene con el tropismo a alfa-2,6 humano. De tal forma que entonces todos estos casos que hemos detectado pues nos pueden, bueno, son asociados a los brotes de influenza aviar en México. Si se fijan, hay tres subtipos aviares predominantes, los H7N3, H5N2 y H5N1, y de cada uno de esos brotes hemos podido detectar un caso o algunos casos en humano de tal forma que está sugiriendo pues la adaptación gradual a humanos. Afortunadamente la transmisión humano-humano no ha sido eficiente, pero tenemos que seguir haciendo los estudios. Por último, el monitoreo extensivo en animales, en medio ambiente, en humanos, es necesario, ¿no? Creo que aquí falta más monitoreo en México, en tanto en ganado como en aves, en el norte del país, para poder seguir detectando H5N1 en nuestro país. Y por último, agradecimientos a mi laboratorio de que estamos trabajando con la calibración molecular de influenza y las colaboraciones que hemos tenido a través de estos años, incluyendo al doctor Darwin Cobasa allá en Canadá, en el Laboratorio Nacional de Microbiología. Gracias.
Thank you, Dr. Perez, for the excellent presentation. Very interesting. I think we have a few, maybe time for one question. I don't see any questions on the chat. Susanna, do you see any questions? I don't.
No, I don't see any question, but I have one question. Can I?
Okay, sure.
Doctor Joel, muchísimas gracias por su presentación, bastante interesante. Como bien lo menciona, creo que hace falta un poquito o refortalecer los sistemas de vigilancia, ¿no? Entonces, no sé si nos podría ahondar más en cómo podríamos mejorar estos sistemas para salvaguardar la salud animal y la salud humana.
Sí, creo que a pesar de que hay poco monitoreo activo, ¿no? Porque creo que esa es la desventaja en México de reaccionar ante algunos brotes de influenza aviar. eh, y a pesar de eso, se han detectado algunos casos en humano. Si cambiamos una búsqueda, ¿no? activa en el país, creo que podemos incrementar la detección de los virus de influenza aviar, tanto en aves como en mamíferos, y creo que podríamos también detectar más casos, al menos de forma serológica, de los casos de virus aviar en humanos. Creo que esa sería mi sugerencia, ¿no? No reaccionar solamente a cuando hay un brote, sino que de forma activa tenemos que tener mayor capacidad para hacer este monitoreo.
Thank you, Dr. Perez. I think that wraps up the morning session. And I want to thank all the 4 speakers this morning talking about avian influenza, improving our knowledge and bringing up the risks and trying to find solutions. And as we mentioned at the beginning, there's funding available for collaborative research between the 3 countries. So I suggest looking into some research collaborations between the countries to address the issues with the avian influenza and the zoonotic risks that we pose. So we will stop here the morning session, and we have a 10 minutes health break, and we will see you all in 10 minutes to discuss about the new world screwworm issues in the Americas. Thank you very much. Hi, Dr. Lomayer. How are you?
I'm well. How are you?
I'm good. See, hope everybody's back. Are you ready?
Yes, I can go ahead and start sharing.
Perfect. Welcome, everyone. So I think we should get started. So this is the afternoon session on the second day. And the afternoon session is dedicated to discuss the new issue that we are all facing, an re-emerging issue in North America related to the screwworms. And the first speaker is Dr. Kim Lomayer from USDA, talking about re-emergence of of screwworm and what has been done in the USDA ARS research group. Dr. Lomayer.
Yes, thank you for the invitation. It's my pleasure to be here today and talk to you about what USDA ARS is doing on the New World Screwworm research front. Just want to make sure you can see my slides. Okay.
Yes, we can.
Okay, great. So I think a lot of us are probably familiar with New World Screwworm, but just in case anyone is not, I'll just briefly introduce it. So what exactly is New World Screwworm and why are we talking about it today? This is an old pest we haven't had to think about literally in decades here in the Americas. Screwworm is a large fly. Its scientific name is Cochleomya omnivorax. It's closely related to other blowflies. It's an obligate parasite of warm-blooded animals, including humans. So it's here at USDA ARS, we focus on control on the livestock and wildlife side, but it is should also be noted that it is a pest of humans as well. It causes what we call myiasis, which is just another word for infestation of living tissue by a fly maggot. And the damage is caused primarily by the feeding of the fly larvae, which is a little bit different than how we usually think about flies. It's usually the adult flies that we worry about taking blood meals and causing health and disease damage. But in this case, it's actually the larvae of these flies. If it goes untreated, it can cause death of its host. And that's not just because of damage from the feeding, which of course, that can be an issue if the infestation numbers are large enough. But it's usually caused by secondary infections that happen because of the size of the wound and the damage to the tissue. You get bacteria and sepsis and other things like that happening in the animals. And this fly is endemic to the Americas. We think it was originally from South America. Just a little bit about the life cycle, because this pest is so unique and a little different than what we've had to think about for a long time. Of course, adult flies find each other and mate. Gravid female then looks for a mammal or a warm-blooded animal with a wound. And these wounds don't have to be very big. It can be something as small as a lesion left even by, say, a tick bite. And if we're all familiar with what those look like, they're pretty small. So that doesn't take much of a wound. So the gravid female finds that wound and lays her eggs. Those eggs hatch pretty quickly within 12 to 24 hours, turning into early stage larvae, L1 and L2 larvae, which then go on to develop into larger larvae, the L3 larvae, within 5 to 7 days. Those L3 larvae then leave the wound and leave the host or the orifice and buries itself in the ground where it pupates. And it spends about 6 to 8 days in the soil as pupae, and then the adult fly emerges and the whole life cycle starts all over again. So if you've ever heard of the sterile insect technique, or SIT, it was actually thought up back in the 1930s because of New World Screwworm. And it takes advantage of the fact that the female screwworms only mate once. So when she's ready to mate, she'll find a male. She only mates once, and then only lays eggs that developed from mating with that one male. So the scientists that came up with sterile insect technique, and I'll mention them here in just a second, took advantage of this caveat of the life cycle. And we've relied on that sterile insect technique for decades now to keep screwworms eradicated from the United States, Mexico, and Central America. So USDA ARS has a very long history of working on New World screwworm. The lab in Kerrville, Texas, is actually named after the two pioneers in New World screwworm work, and the two gentlemen who came up with the idea of sterile insect technique and put it to good use against New World screwworm. And those are Dr. Nippling and Dr. Bushland, featured here. That's Dr. Bushland with the red circle around his face, and Dr. Nippling with the blue circle around his face. This picture was actually taken in Menard, Texas, which is about an hour-ish from where I am here in South Texas, back in the 1930s. So USDA back then had several locations that were working on screwworm. It was such an important pest in Texas and in the United States back in that time. And Dr. Nippling and Bushland, that's where they first met, working on screwworm. So then in the late 1930s, that's when Dr. Nippling developed this concept of the sterile male technique or sterile insect technique. Late 30s to mid 1940s, though, this was forgotten about a little bit, or maybe not forgotten about, let's say it was shelved, because Dr. Nippling and other scientists working in the United States and at the lab in Menard and across the nation were pulled away to focus their research efforts in support of World War II. So it wasn't until the mid 1940s, around 1946, all of these labs that had been working on screwworm, there was one in Dallas, Texas, Uvalde, Texas, and Menard, that were consolidated here into Kerr County in the mid 1940s, put them all in one place to work on screwworm. In the early 1950s, Dr. Bushland produced the first viable sterile male screwworms. And then in the 1960s, our laboratory was moved here to our current location in Kerrville. We've been here over 75 years now. And then, of course, that the story picks up from there, the one we're all more familiar with. Between the 1950s and the 1970s, screwworms are eradicated from the United States. So here's just a pictorial timeline of how that eradication process worked. The early 1950s were when the first males, viable sterile males, were produced. Between the 50s and the 70s, the screwworms are eradicated from the U.S. By the 1980s, they'd been eradicated from Mexico and pushed down into Central America. And then between 1997 and the early 2000s, screwworms are eradicated from Central America and Panama. And they put into effect what we called a quarantine line there in the Darien Gap in Panama. So if you're familiar with that part of the world, that Darien Gap was a nice line to maintain quarantine and eradication that separates Panama from Colombia. Today, that quarantine line is maintained with flies that are produced in Pecora, Panama. This is COPEG, which is the sterile screwworm production facility. It's a Panama U.S. Commission for the Eradication and Prevention of Screwworm. That's where that COPEG acronym comes from. Again, located in Pecora, Panama. It's been operational since 2006. Before 2006, all of the sterile male flies that were produced as part of this eradication effort were produced either in the United States or Mexico. Once they were eradicated from Mexico, then the production facility was moved and maintained there in Pecora. And up until last year, this facility produced between 20 and 40 million flies per week. With the recent reemergence of New World screwworm back into Central America and Mexico, the Pecora facility is now producing about 110 million flies a week, and it's at their maximum production capacity based on the size of this facility. All of those flies are being diverted to the front line now to try to slow down the northern progress of New World screwworm through Central America and Mexico. So should screwworms get back into the United States, this graph gives a little bit of information about what those estimated losses would be. And this was recently updated by our colleagues at APHIS. And this looks just at Texas. So it's a pretty staggering figure. Just at Texas alone, cattle producer losses would be estimated at over at almost $2 billion a year just on the cattle side. This, of course, does not take into consideration losses associated with wildlife, which if you're familiar with how ranching is done in Texas now, not only do we ranch cattle, but we also ranch wildlife like white-tailed deer, but exotics like nilgai antelope, black buck, other exotics like that as well, very much contribute to the Texas economy and are almost as popular in some locations as cattle production for a way to make a living. So where does USDA ARS fit into what's going on with New World screwworm? Well, as I mentioned, we've never stopped working on screwworm. It's just, of course, things have ramped up in the last 2 years. APHIS, the regulatory work that APHIS does makes them our primary stakeholder when it comes to the research we do on New World screwworm, as well, of course, as our U.S. beef and wildlife producers are primary stakeholders in these efforts as well. We currently have 3 scientists and 1 rapid response postdoc involved in our New World screwworm research program. And we keep 1 research scientist stationed in Pecora, Panama at COPEG at all times. And with this being a quarantine species, we don't actively rear this pest in the United States. There is no entity, either federal or academic or otherwise, that maintains a colony of New World screwworm in the U.S. So any work we do with live screwworms has to be done down at the Pecora facility. So we do keep someone from ARS stationed down there at all times. We rotate them in and out about every 3 to 4 years. As soon as one comes back, we send the next person in line down to run that research program on the ARS side. We solely focus on research priorities that we identify with APHIS to meet screwworm production, regulatory, and mitigation needs. We meet at least weekly, sometimes daily with APHIS to ensure that the research priorities we have in mind meet the needs that they have on the regulatory side. And of course, any work with live New World screwworm is done in coordination and cooperation with our colleagues at the COPEG facility. So we look at this as a 4-pronged integrated research approach that includes modeling, education, surveillance, and management. On the modeling side, we are looking at where and when to survey, and then how to manage that infestation data that we have, tools to use that data and that information to come up with predictive tools that APHIS and that our producers can use for determining when and where screwworm might be a problem in the United States. Education is a big part of this. I'll go into detail about that more in a minute, but that, of course, includes training. And surveillance is a huge part of the research that we are doing, how we can better detect New World screwworm introductions and monitor treatments, and then, of course, management. And we need new tools in our toolbox to either eliminate or at least better manage New World screwworm. So I'll talk about each of these individually. So we're doing a lot of work with modeling. We have our scientist in Pecora, Panama, Mackenzie Tejan, is working on some modeling work, but a lot of this we are doing in collaboration with our ARS partners at NBAF in Kansas, and these efforts are being led by Dr. Lee Kohnstedt. And so as we look at the United States and Mexico, Central America, but particularly the United States, and trying to predict where screwworms may re-enter the country if they continue to move northward, and how we could be better prepared for that. This also all takes into, of course, looking at current climate across the southwestern United States. We are, of course, trying to predict where these flies are going to show up and how badly they may show up, right? So some of the work Dr. Kodstead and his group have been doing are looking at what we would call the vulnerable stages of New World screwworm to the environment. So that would be the pupal stage, the adult stage, and the egg stage. So really any stage that's not actively in a wound in a host is vulnerable to the climate. So very hot temperatures, very cold temperatures, those are going to be the limiting factor of where in the United States moving forward that would have the potential to be good areas where we might see New World screwworm showing back up and thriving. So you can kind of see some of this modeling work here reflected on the screen. These slides were made by Dr. Chad Fott that are a part of this modeling group. So they did this work by going in and putting the parameters that we know are limiting factors on these life stages, and then inputting current weather data. That black line that you see moving across that you see drawn shows the old limits of where we found screwworms when they were in the United States before they were eradicated. And so you can kind of see with current climate data that we could put into this model that we may actually see if New World screwworm gets back into the United States, extending beyond that old limiting range that we used to see in the 40s and 50s here in the U.S. Dr. Konstad's group is also looking at using seasonality to be able to predict seasonality of risk at any location. And this slide shows just an example. We can come in and pick a spot in Texas or anywhere else in the United States with up to a square kilometer of resolution. And we can, do this even on a county level. And we can go in and pick that spot, and you can see then if we look at day of the year, it'll tell us the suitability index for that particular area. So you can kind of see here in looking at this that where we're seeing these screwworms may vary a little bit from year to year, but there is a general flow, if you will, a general timeline of where these would be most likely to occur in any given spot at any time of the year. And this, of course, would be helpful as we look at some of our southwestern states that are adjacent to Mexico in trying to predict what areas are going to need the greatest level of surveillance if and when these flies start to move north. This is another slide that reflects some of the modeling products that the modeling group is working on. We have an agent-based model for seasonal suitability for New World screwworm, a graph neural network, cellular automation, nowcast, and they have a few other models that they're working on as well. So final products to come out of this modeling work would be spatiotemporal predictions for U.S. entry. And what you're seeing reflected in this timeline slide to your right is they're taking this case data that we have and weather data that we have on New World screwworm and overlaying that with the areas where we are most likely to see the greatest density of cattle hosts. So as you can see, it kind of gives us a little bit of a scary picture maybe, but also some really good information on what areas we need to be looking at as the greatest potential threat for where New World screwworm might emerge and do best in the U.S. should it reach us again. And of course, this information comes in handy for providing direction and for testing of management strategies. These can be used to do predictive work and make protocol suggestions for where best to concentrate surveillance and management efforts. The second tier of this that I wanted to talk about is education. And because New World screwworm had not been in Mexico and most of Central America and all of the United States for decades, it really is something that we haven't had to worry about as a population for a very long time. I ran into very few people that have any experience with New World screwworm, and if they do, they're at an age now that they were likely very young children when screwworm was in the United States before. So they remember riding along with a parent or a family member to scout cattle and to treat New World screwworm, but it's not something the average person has had to be aware of for a very long time. So we're finding that education is key in all of this. It's something that's really helped countries in Central America to try to get ahead of this, be aware of what they're looking at, and to start thinking about good animal husbandry practices as well as possible treatment methods. So USDA, ARS, and APHIS, with our animal health partners, have been spending a good amount of time doing outreach and doing education on New World screwworm. And these are some photos from some trainings that we have done here in Texas, informational seminars on getting the word out. And we've also been doing a lot of hands-on training with inspectors from both APHIS as well as the Texas Animal Health Commission to have boots on the ground here in Texas and through some other southwestern and southeastern countries, having people prepared should screwworms reach the United States, how to do trapping, how to do identification, how to do surveillance. Part of this educational process is the development of New World screwworm educational materials as well as reference collections. We've been working with our partners at COPEG to put together specimens of larvae and adults of New World screwworm to send out reference collections to universities and federal partners that would like to have these insects in hand for training their staff for identification. We've also put together educational materials like what you're seeing on the screen to help people be trained to know the difference in identification of screwworm in comparison with other blowflies like secondary screwworm. As you can see from these photos, at first glance, these can be quite difficult to tell apart. New World screwworm looks very similar to secondary screwworm, as I have reflected here in pictures of both. Secondary screwworm is very common here in the United States. So as you can imagine, when you compare these two, that producers or citizens out that are seeing these, if you don't know what you're looking for, this could cause a bit of a panic. So having people trained or having these materials available for education have become extremely important. Surveillance, on the surveillance side, ARS is doing a lot of research to improve our surveillance capabilities. We're working to develop novel attractants and lures for screwworms. And as part of this, we're doing work to identify volatile and non-volatile pheromones, particular hydrocarbons, things like that these insects use to cue in on finding each other or finding their hosts. Looking at things like mating behaviors as well, because we think that plays an important role in being able to successfully lure and trap these insects. There are lures out there that work to attract New World screwworm as well as other blowflies, but the problem is we get things other than screwworms coming into those traps. And this picture up at the top right is an example, and that's probably from the 1930s, 1940s. Those are all flies in that pile, not all screwworms, but they were trying to attract for screwworms using attractants like rotting liver or a dead animal. And as you can see, you not only get screwworms, you get a lot of other flies as well. So we think we can do that better. ARS is actively working on an improved lure, our goal being to develop one that is attractive specifically for female New World screwworm. So if we can only attract females, that helps us with, one, knowing those females are out there, two, pulling those mating females out of the environment, and three, if we are releasing sterile male flies in those locations, then we aren't pulling those sterile male flies out of the environment as well. This is Dr. Paul Hickner and Dr. Zamira Youssef. They are actively doing this chemical ecology work to try to develop this improved lure for female New World screwworms. So part of that, if we get a lure that works really well, we want to make sure we have a trap that works really well as well. There are fly traps out there that work for New World screwworm, but again, ARS is looking to actively improve those. So we're looking at things to couple this improved lure with an improved trap, which would enable us to then be able to have what we would call a trap kill system, that we can put these traps out to do surveillance, but also to actively pull these gravid females from the environment and help the regulatory program by doing that. We're doing quite a bit of work on the screwworm management side of things. We do a lot of work to try to help improve the sterile insect technique when it comes to screwworms. So we're looking at things like rearing optimization, strain health monitoring. So we call it, how can we build and rear a better fly? So when we are producing those millions of flies a week at COPEG or at other potential production facilities that may be in the works, how can we get the most fly for our money and for our effort, if you will? As part of that, we're exploring screwworm microbial ecology. If we can take advantage or improve the natural microbial ecology of the fly, that might enable us to either take advantage of some control mechanisms there, or to eliminate things like bacteria that may be growing in the plant. We actively now use formaldehyde in the rearing process, which is not much fun for the staff to have to work with. So if we can come up with better preservatives to keep these microbial agents from growing in the production flies, that would be helpful. We're also looking at things like genome assembly and population genetics. The more we know about the genome of these flies and the better assemblies that we have, again, helps us to rear a better fly, but it also helps us to find segments of the genome of the animal that might be potential targets for control, looking at things like vaccines and other novel control mechanisms like that. We're also looking at rearing waste management. When you produce hundreds of millions of flies a week, you also produce thousands of liters as waste associated with that, especially when you're feeding them a diet that's made up of animal components like red blood cells and plasma. So we're looking, always looking for better ways to manage the waste that's produced by the rearing process. APHIS has identified to us that a mark and recapture system would be very helpful for them in the field. So if we're catching flies out in the field, how do we know if those are our production flies or our wild flies? So that is something we're actively working on, is coming up with a easy to use, safe and inexpensive mark and recapture system. And we're also have developed a genetically engineered male-only strain of the fly. This work was done in collaboration with Dr. Max Scott from North Carolina State University's lab. So it's a female lethal, all male candidate strain, and it utilizes a single insertion with both off and on and lethality components. So it's a TET off model. So if this strain could be put into production, it would be easier to ID and screen and cross into different backgrounds. And so this involves a new construct gene with promoters and on and off switches and a fluorescent marker from another arthropod organism that's being inserted. So it's not an actual modification of the existing screwworm gene. It does very well so far on mass rearing diets with performance equal to the current production strain. If it were to get released fertile, all offspring would be male carriers, so that makes us more confident about widespread use of this organism in the field. And what we really like about this is it would double the number of sterile male flies that could be produced each week. So when I mentioned earlier that the COPEG facility makes 110 million flies a week right now, that's actually only half of that are sterile males. The other half are sterile females. We don't currently have a way of sexing these flies easily and quickly. So we produce both sterile male and female flies each week. If at some point we could transfer to this all male strain, that would allow us to allow APHIS to produce a true 110 million a week, or whatever number that is we're shooting for a week, that are all male. So long story short, would double our capacity. And we are in active discussions with the USDA regulatory groups and EPA at this point. There is a lot of interest to try to get this all male strain up and going. So we are putting a lot of efforts toward trying to get this through the regulatory process. Also on the management side, we are looking for SIT additions, so things that we can use to complement the sterile insect technique efforts. We are looking for pesticides that we can use in the United States. Right now, there are literally only 3 things that are labeled for use for animals infested with New World screwworm in the United States. Again, this goes back to this being a pest that we haven't had to worry about in about 50 to 60 years. So there are 2 permethrin sprays, wound sprays, that are labeled for use against screwworms right now. So this is a spray that's got a wound drying agent in it, as well as permethrin that can be used here in the U.S. And then also a flowable formulation of Kumafos, but this it's labeled for use against screwworm, but because it's an OP, it can only be used as part of under regulatory supervision by APHIS right now. So more tools in the toolbox are needed. We're actively working with FDA to look at other insecticides that are labeled for use on other arthropod pests that might be easily, that we might be able to get emergency use exemption for to use on cattle here in the United States. Also looking at compounds from other countries that are actively used against screwworm that might be possibly able to be used here in the United States. We're starting to do some work on resistance monitoring because if these get into the United States, we want to know where they fall out resistance-wise. Hopefully they're not resistant to anything at this time, but that's something we need to be looking at. We're also looking at possible use of a parasitoid for trying to help add to the control program for New World screwworm. And then I was going to mention FDA recently did get approval to start considering some of these compounds for emergency use. And with that, here is my contact information. Modeling questions can be directed to my colleague, ARS colleague at NBAF, Dr. Lee Konstead, and there is his email address as well.
Thank you, Dr. Lemire. Thank you for the nice presentation, great introduction to screwworm and the and what's going on in the United States and all the programs that you have. I think due to the time constraint that we're going to move to the next speaker, and you can answer any questions that's on the chat later.
Okay,
and the next speaker is Dr. Ramiro from the National Services of Agri-Food Health, Safety and Quality from Mexico, talking about screwworm control and containment strategies in Mexico. Dr. Ramiro, the floor is yours. Thank you.
Gracias. Muy buenas tardes a todos. Bueno, el tema que vamos a tratar en este momento es las estrategias de control que se están llevando a cabo en México por parte del SENASICA para gusano barrenador del ganado. Y bueno, es muy importante considerar algunos aspectos que acaba de mencionar la doctora en la ponencia anterior, específicamente del parásito, de la mosca. Eso es importante porque de esta manera podemos tener algunas estrategias con mejor eficacia, eficiencia para el control de GBG. Siempre es importante considerar algunos aspectos como son los retos a los que nos enfrentamos. Si bien es cierto, es un problema que en México está causando graves problemas en el sur, en el sureste del país, hay algunos aspectos que nosotros llamamos determinantes sociales que complican las acciones de control en este sentido. Entonces, debemos de considerar los retos a los que nos enfrentamos y unos de ellos son principalmente el hecho de que existan movilidad, por ejemplo. También importante considerar que este parásito que podemos ver aquí en la imagen tiene un ciclo biológico bastante acelerado. Su reproducción es exponencial, entonces eso implica la reproducción constante y acelerada de este parásito. El otro reto al cual nos enfrentamos es que es una enfermedad, un problema que afecta no solamente a una especie, son varias especies las que se ven afectadas. Todos los animales de sangre caliente, incluyendo animales silvestres, se pueden ver afectados. La movilidad humana y la movilidad o la movilización de animales también juega un papel determinante en la diseminación de este problema. La pobreza y accesos a servicios de salud definitivamente son un factor que influye en la diseminación de este problema, de este parásito. Otra situación que debemos de considerar es las prácticas ganaderas que se llevan a cabo en México, seguramente en otros países también, pero en México hay diversas prácticas que se llevan a cabo, como por ejemplo el aretado de los animales, el marcaje con hierro, el manejo incluso de los animales que predispone a los animales a que tengan heridas, a que sufran lesiones, y eso obviamente es altamente susceptible para una infestación por este parásito. Otra de las limitantes que tenemos y los retos a los que nos enfrentamos es la parte de conciencia y educación sociosanitaria. Aunque se han realizado bastantes actividades en México de conciencia y de educación sociosanitaria, es complejo. Hay gente que en algunos momentos se rehúsa a hacer el reporte o se rehúsa a realizar acciones de control de este parásito. En conjunto, todos estos factores que acabo de mencionar son la combinación perfecta para que el entorno sea propicio para la proliferación del gusano barrenador del ganado. Obviamente esto afectando no solamente a la salud animal, también a la salud pública, la ganadería y por consecuencia a la seguridad alimentaria. Eso solamente es mencionar a todos los retos que nos enfrentamos y con lo que convivimos día a día aquí en las estrategias que se tienen por Senacica para el control de gusano barrenador del ganado. Obviamente este problema no se tiene que atacar de una sola vía, o sea, tiene que ser un ataque multifactorial. Obviamente se trabaja bajo el enfoque de una salud. El título que se tiene en este taller de riesgos compartidos, soluciones compartidas, me parece muy adecuado. Justo eso es lo que se requiere para poder abordar este problema de gusano barrenador del ganado, porque finalmente es una enfermedad transfronteriza. ¿Sí? Que si un país puede realizar acciones, pero el país vecino no las realiza, pues definitivamente no se puede avanzar en el control de este problema. Además, no solamente entre países, sino a nivel país, se tiene que abordar desde tres puntos importantes, que es el eje para abordar con el enfoque de una salud, agricultura, en este caso que ve el tema de salud animal, medio ambiente, que por consecuencia ve la parte de animales silvestres, y la Secretaría de Salud, que es todo lo que tiene que ver con las afecciones que esto puede traer a la salud pública. En México se trabaja muy estrechamente con las tres secretarías, tanto salud como medio ambiente trabajan de la mano con agricultura a través de Senacica para realizar acciones en conjunto y poder abordar este problema desde estos tres puntos importantes. Quiero comentarles que la situación de gusano barrenador del ganado en México se presentó el primer caso desde el año pasado, el 21 de noviembre, para ser exactos, en un punto de inspección federal, en donde se revisan todos los animales como estrategia de este control que se está realizando o de prevención. Se tenía la inspección de animales en los puntos de inspección federal y un cargamento que procedía del sur del país se dirigía hacia el centro del país y se detectó un animal con gusanera en una oreja. Ese fue el primer caso que se detectó en noviembre del año pasado. A esta fecha que acabo de mencionar, estamos con 6,000 casos confirmados. Es un total de casos acumulados hasta este momento, son 6,000 casos. Como pueden observar ahí, son 7 estados que se han visto afectados, principalmente son los estados del sur-sureste de nuestro país. Podemos observar ahí que son 298 municipios en los cuales se ha tenido presencia de este problema, de esta parasitosis. El primer lugar lo ocupa el estado de Chiapas con más de 3,000 casos. En segundo lugar, Tabasco, que es un estado fronterizo con Guatemala. Chiapas también lo es. Campeche es un estado fronterizo con 447 casos acumulados. Le sigue Quintana Roo, Oaxaca, Veracruz, que no son estados fronterizos, pero el incremento de casos se ha dado por las condiciones también climatológicas y obviamente de ganadería que se presentan en estos estados. Ahora, las estrategias que se tienen implementadas en México a través del SENACICA están relacionadas con el Dispositivo Nacional de Emergencia en Sanidad Animal, que se activó desde el año pasado para realizar acciones extraordinarias preventivas en su momento para, pues obviamente mitigar el riesgo de que el problema pudiera llegar a México. Sin embargo, pues lo acabo de mencionar, llegó a nuestro país en noviembre del año pasado y las estrategias que se tienen fuertemente son principalmente la promoción de la notificación y la educación sanitaria. Eso es nuestro mejor aliado, es la única manera que podemos llegar a cualquier sitio y lo hemos hecho de gran manera. Sin embargo, comento que, bueno, pues hemos tenido dificultades para llegar a cada uno de los rincones del país, pero sí se están realizando diversas acciones en este sentido y es lo que ha permitido que se den esas notificaciones y que se atiendan los casos. Apostamos mucho a esto porque es la única manera de que si los productores están conscientes de que existe el problema, de que qué tienen que hacer con los animales, cómo prevenirlos y en caso de detectarlos, a dónde reportarlos, nosotros estamos un paso adelante. El control de la movilización es otra estrategia bastante importante que se está realizando también en nuestro país. Es un problema bastante complejo. La atención oportuna de casos y los tratamientos. Otro de los aspectos importantes es el diagnóstico a distancia que estamos realizando, lo que nos ha permitido la toma de decisiones de manera rápida, precoz, al detectar casos en los puntos de inspección o asimismo en algunos ranchos ganaderos. La dispersión de mosca estéril es un complemento a las acciones terrestres que se llevan en los ranchos y en todas las unidades de producción pecuaria. Asimismo, se realizan acciones de desinfestación de instalaciones donde se han manejado esos animales que han sido positivos, que tienen miasis por gusano barrenador del ganado, y el seguimiento de estos casos, la vigilancia e investigación epidemiológica, que son parte importante para poder dar trazabilidad a los casos y predecir futuros casos que pudieran presentarse. Voy a ir tocando tema por tema de manera breve para no extenderme con el tiempo que tengo asignado. Quiero comentar que dentro de las actividades de promoción de la notificación y educación sanitaria se han realizado distintas pláticas, no solo a nivel de médicos veterinarios, se han realizado a nivel de productores, amas de casa, a nivel de servicios de salud, en todos los medios, en todos los niveles hemos hecho presencia con la intención de poder llegar con el mensaje y entregar no solamente el mensaje, sino también a estas personas que participan se les entregan tubos para la colecta de muestras, se les entrega material impreso para que tengan la información presente en cualquier momento. Se hace uso de diversos medios, no solamente los medios impresos, lonas que podemos observar ahí que se colocan en varios lugares, carteles, trípticos, material digital que se distribuye a través de las redes sociales. Se ha participado con las asociaciones ganaderas, con apoyo de las organizaciones de productores, la Confederación Nacional de Organizaciones Ganaderas y otras instituciones, principalmente también instituciones educativas, que nos han permitido acercarnos a diversos actores del sector pecuario. Solo por mencionar, se han realizado 2.271 pláticas del año pasado y lo que va de este año, con más de 87,000 participantes que han sido concientizados en este aspecto. Comentaba la distribución de kits para la colecta de muestras, que es algo importante que nosotros nos permitimos hacer llegar de manera gratuita a cualquier persona, no solamente ganaderos, incluso personas poseedores de mascotas o de otro tipo de especies productivas, con la intención de que ellos en el momento que detecten una miasis puedan colectar las muestras o algún médico veterinario colecte las muestras en lo que el personal se puede trasladar al sitio para poder dar la atención complementaria. En estos kits que nosotros repartimos de manera gratuita, se otorga un tubo con el medio que es alcohol al 70%, las pinzas para poder colectar las muestras, un sobre con un polvo que es el polvo curativo y que además tiene efecto contra el gusano barrenador del ganado, es muy efectivo y además permite acelerar la cicatrización de la herida y un formato donde vienen los datos principales que debe de contener para poder registrar el caso y darle seguimiento. Estos tubos se han distribuido a todos los niveles y se han distribuido más de 169,000 de estos kits que, bueno, pues finalmente hemos tenido buenos resultados con esto. Obviamente el sobre también les funciona para que en caso de que tengan animales heridos lo puedan aplicar y prevenir alguna infestación por gusano barrenador del ganado. Además de eso, parte también de la educación zoosanitaria que tenemos es un curso en línea que se ha ofrecido a estudiantes principalmente, profesionales involucrados en las actividades pecuarias, que el curso finalmente nosotros lo teníamos disponible desde marzo del 2023. Sin embargo, aquí podemos hacer una mención que desde que se echó a andar este curso, desde que se puso en marcha, se han impartido 187 cursos con más de 118 instituciones que han participado y más de 16,000 que han cursado este curso en línea y que de alguna manera también es una acción de educación que se tiene con bastante demanda. Dentro de la capacitación también preparamos a grupos de respuesta a través de simulacros de gabinete y de campo que se realizan en diversas entidades del país. Estos cursos, estas capacitaciones para preparar a los grupos de respuesta se realizan principalmente en zonas donde no se han presentado los casos de gusano barrenador del ganado, principalmente en la parte norte y centro del país. Los primeros 2 simulacros que se realizaron fueron en Chiapas antes de que ingresara el problema a nuestro país. Posteriormente se han dado estos cursos y tenemos en puerta más cursos que sirven también para esa integración con las autoridades involucradas en cada estado para poder abordar en caso de que se presente en cada una de las entidades de nuestro país. Estos grupos se preparan de manera teórica también, pero hay una práctica en donde ellos permiten observar larvas, identificar prácticamente el gusano, no solamente para que ellos hagan el diagnóstico, de hecho esa no es la finalidad, sino para que participen con acciones con nosotros y además la manera de inspeccionar animales, tratarlos y aplicar las acciones contraepidémicas que estamos realizando en Senacica. La atención que damos a través del reporte que hacen los productores es muy importante para nosotros. Esta es la manera, podemos observar ahí algunas imágenes de los compañeros que se encuentran en campo realizando estas acciones tan importantes para reducir la cantidad de moscas que se encuentran presentes en el terreno prácticamente. Y esto lo hacemos una vez que nos notifican, nosotros acudimos, el personal de campo acude a las instalaciones, a los predios afectados, a revisar el animal enfermo, que hasta el momento la mayor parte de las notificaciones reportan 1 o máximo 2 animales con miasis. No es una cantidad importante de animales que se vean afectados, solamente es 1 o 2 principalmente los que se reportan. Se revisan estos animales, se extraen las larvas, se hace la curación. Una vez que ya se extrajeron todas las larvas de la herida, se hace la curación, se aplica el producto que estamos utilizando que tiene un efecto larvicida y que está funcionando muy bien, es muy efectivo. Además de eso se aplica un antiparasitario y a su vez se aplica un baño de aspersión, no solamente al animal afectado, sino a todos los animales que se encuentran en ese predio, en ese rancho, se revisan y además de revisarlo se aplica el baño de aspersión. Una vez que se concluye con la revisión de los animales, el baño y la aplicación del antiparasitario, se realiza una remoción del terreno en donde se manejó el animal y donde estuvo en su mayor parte el animal para evitar que si alguna larva cayó al piso pueda continuar con el ciclo biológico, porque sabemos la reproducción tan acelerada que tiene este parásito. Entonces, esa es una de las actividades más importantes que se tienen para disminuir el número de moscas presentes en los predios. Les había comentado que el diagnóstico a distancia o telediagnóstico lo utilizamos de manera que podemos acortar el diagnóstico, la emisión de un resultado del laboratorio oficial que tenemos, que actualmente es el CENAPA, el que se encuentra en Jiutepec, Morelos, aquí en nuestro país, y se está extendiendo a la red de laboratorios que tiene también el Senacica en todo el país para poder corroborar los diagnósticos que están realizando. Los compañeros que se encuentran en campo, cada uno de ellos porta un microscopio, pues prácticamente es sencillo el microscopio, pero permite obtener imágenes con buena definición, que estas imágenes pueden ser retransmitidas al laboratorio para que los especialistas realicen la identificación taxonómica con base a las estructuras que se están proyectando en estas imágenes y pueda determinar la especie de la que se trata, confirmar o descartar casos de gusano barrenador del ganado, y eso nos permite tomar acciones de manera inmediata directamente en el predio donde nos estamos encontrando. Además de estas acciones que se realizan, es importante comentar que a través de los organismos auxiliares de sanidad animal se realizan acciones, además de la vigilancia pasiva, que es que los médicos vayan a los sitios donde reportan que hay presencia de animales con gusaneras, con miasis. Se hacen acciones en sitios donde se van identificando casos, sobre todo los casos que están más al norte, de donde se están presentando la mayor parte de los casos, y se realizan acciones de vigilancia activa. Esto quiere decir que los compañeros visitan los ranchos, revisan cada uno de los animales, inspeccionan que no tengan heridas, si encuentran heridas, las curan con el producto que estamos utilizando, y si detectaran alguna miasis, en ese momento se realizan las acciones que acabo de mencionar anteriormente. Hasta el momento, esta información que tenemos reportada el 20 de agosto, se habían revisado más de 11,000 ranchos, unidades de producción ganaderas, y se habían inspeccionado más de 85,000 animales, de los cuales por lo menos 41,000 animales han sido tratados, o sea, curados, porque tenían alguna lesión, que bueno, eso finalmente pues es atractivo para la mosca y es importante que sea curado. A su vez, se les da la capacitación a los productores para que ellos realicen la curación y prevengan. Como es un problema que anteriormente no estaba presente ya en nuestro país, fue erradicado, la gente no estaba acostumbrada a realizar acciones de curación de ombligos, de tratamiento de lesiones que se encontraran en los animales, y actualmente esas acciones tienen que ser más frecuentes, tienen que revisar sus animales y entonces se les explica cómo realizar esas acciones. Es muy dramático ver los casos en campo porque con 2, 3 días de que la mosca llegue, de que tenga una herida, los animales se ven severamente lastimados, lesionados y bueno, pues obviamente eso puede comprometer la vida de los animales. Las acciones que se realizan para el control de la movilización están a cargo de la Dirección General de Inspección Fitosanitaria. Entonces, se tienen dos barreras originalmente planeadas desde el año pasado, que son las que podemos ver en la parte sur de nuestro país. Estaba conformada por Huixla, Trinitaria y Catazajá, son los 3 puntos, así se denominan, que realizaban acciones de inspección al 100% de los animales que cruzaban por estos sitios. Como el problema arrebasó estos puntos, pues se pusieron en marcha otros puntos, que son los que podemos ver con línea azul, y estos puntos realizan las mismas acciones. Posteriormente se tiene una tercera barrera que está realizando acciones también de inspección y verificación, aunque no tiene todavía la habilidad de poder realizar la inspección del 100% de los animales. Se inspeccionan por fuera de los medios de transporte, se realiza la inspección documental de los animales que están siendo trasladados a otras partes del país y que, bueno, finalmente, pues es acción que puede mitigar el riesgo de desplazamiento de animales que pudieran verse infestados por mosca de gusano barrenador del ganado. Obviamente, estas acciones no son las únicas que tienen que estar, todas tienen que ir de la mano. Si los productores, los comercializadores, los transportistas están conscientes del problema, no van a movilizar animales infestados. Si esto lo desconocen, pues seguramente esto puede propiciar un desplazamiento de animales en esas condiciones, que obviamente pues es dañino para el país. El control de la movilización que realiza en los puntos de inspección federal inicia principalmente, sobre todo en los 3 puntos que había mencionado, y en Catazajá, que es donde tenemos la inspección a través de binomios caninos, que son perros que han sido adiestrados exclusivamente para el tratamiento, para la inspección de los animales, que, bueno, finalmente algunas movilizaciones se realizan por la noche y estas movilizaciones complican obviamente al ojo humano que pueda realizar la inspección a detalle. Por eso es que se hace uso de los binomios caninos, que ellos detectan o pueden detectar alguna infestación o alguna herida que pudiera ser susceptible a miasis y marcan para que posteriormente un inspector, un técnico del Senacica pueda realizar la inspección detallada de ese animal y en caso de detectar alguna herida es curada y si hay presencia de larvas, pues se pueda tomar las muestras. Estas muestras, una vez que se toman, son analizadas con el microscopio a distancia con los expertos que les había comentado. Y si se detecta que es gusano barrenador del ganado, este cargamento no puede seguir su camino. Obviamente se tienen que aplicar algunas acciones de mitigación de riesgo, como es la aplicación de antiparasitario, un baño de aspersión al 100% de los animales que cruzan por estos sitios. Estos cargamentos tienen que flejarse y se les emite una constancia para que puedan continuar en caso de que no tengan animales con miasis. Si tuviera algún animal, alguna presencia de miasis, estos animales son retornados y para eso tenemos el apoyo de la Guardia Nacional, quien en caso de detectarse algún cargamento en estas condiciones, resguarda el cargamento hasta llevarlo a su origen, en donde es obviamente recibido por personal de nosotros de Senacica. Es constatado el descargue de estos animales y se le da seguimiento. Este cargamento debe permanecer 30 días en este sitio hasta que se considere que no representa un riesgo y pueda ser liberado. Podemos ver algunos otros puntos de inspección que no tienen todavía las condiciones de los puntos anteriores que habían mencionado, pero estos puntos tienen el alertamiento para inspeccionar de manera física al exterior de los cargamentos y documental para, obviamente, controlar la movilización. Todos los puntos que podemos observar ahí son puntos, algunos son de inspección federal, algunos son puntos internos, o sea, de cada estado, que realizan la inspección de los cargamentos que pudieran cruzar por estos sitios y, obviamente, evitar que haya alguna evasión de estos cargamentos y que los cargamentos que se movilizan de ganado, pues cuenten con las condiciones para ser movilizados hacia otros estados. Parte complementaria de las acciones de control que se tienen en México es la dispersión de mosca estéril, que se realiza con mosca, que obviamente es una mosca que se produce en la planta de Panamá, ¿sí? Que obviamente con el apoyo del APHIS se está liberando en nuestro país y las parrillas de dispersión se van movilizando de acuerdo a la presencia de casos. Las parrillas tienen que estar adelante de los casos para poder causar el efecto que requerimos. Obviamente, si la cantidad de moscas que se encuentran vía terrestre en campo directamente son mayores a las moscas que están dispersando, definitivamente la técnica no funciona. Entonces tenemos que hacer muchas acciones en campo para disminuir la cantidad de moscas que puedan estar presentes y de ahí la técnica del insecto estéril pueda surtir efecto. La cantidad que tenemos en estos momentos que se está dispersando son 100 millones semanales que se dispersan en México. Aquí podemos observar las parrillas que actualmente están implementadas y las cuales están dispersando mosca. Principalmente es vía aérea. En algún momento se colocaron algunas cámaras de dispersión terrestre, pero es más funcional la dispersión aérea, por lo cual se está realizando de esa manera. Y vuelvo a repetir porque esto ha sido uno de los cuestionamientos más frecuentes que se han dado en diversos foros, que es la mosca estéril. Pero más que la mosca estéril, debemos de apostar a las acciones que se deben realizar en campo con los productores, de reportar, de curar los animales, de evitar que las moscas puedan llegar a infestar sus animales y de esa manera reducir la cantidad de moscas que puedan estar presentes. Porque volví a repetir que es una reproducción acelerada de moscas. Entonces, si nosotros podemos evitar que esto se dé, que la liberación, que la dispersión complemente las acciones de campo, es efectivo. De lo contrario, esto no tiene ningún fin. Entonces, eso sí es importante mencionarlo porque debe de ser un complemento de las dos acciones, además de lo que acabo de mencionar anteriormente, que también son estrategias importantes que se deben de seguir. Adicional a las acciones que acabo de comentar, en México, por parte del Senacica, se realizó una regionalización para definir zonas específicas, que aquí podemos mencionarlas, son 4 zonas. Una zona es la zona afectada, donde se encuentran casos ya de gusano barrenador del ganado, una zona buffer, en donde es próxima a esta zona y que es una zona de amortiguamiento prácticamente, y una zona buffer en la cual existen corrales de inspección. Entonces, es una forma de proteger la parte norte del país. Y el proteger la parte norte del país tiene una funcionalidad. O sea, es poder ir erradicando y controlando el problema del norte al sur, como se ha hecho en años pasados. Entonces, finalmente, todo el país es importante para nosotros. Para México finalmente tiene que considerarse la erradicación completa, pero siempre la erradicación se da del norte hacia el sur. Por eso se tiene esta regionalización establecida y se realizan acciones en cada una de estas zonas. Además de lo anterior, en el norte, a través de la Dirección de Epidemiología de la Dirección General de Salud Animal, se colocaron trampas en el norte del país. Estas trampas tienen la funcionalidad y el objetivo de poder detectar oportunamente la presencia de alguna mosca de gusano barrenador del ganado. Se colocaron más de 1,200 trampas en todos los estados fronterizos de nuestro país, en todo el norte, y podemos observar que estas trampas fueron colocadas estratégicamente, principalmente cerca de centros de acopio, cerca de corrales que se ocupan para exportación, de los puntos de verificación e inspección, en rastros, cerca de rastros, de rutas de transporte de ganado y en algunos tianguis o subastas que se realizan de ganado. Obviamente las trampas son colocadas estratégicamente en zonas donde puede estar presente la mosca. La mosca no va a estar presente donde están los animales. Ahí solamente la mosca busca dónde ovipositar. Ya una vez que la mosca grávida tiene que hacer la oviposición, pero las moscas macho y las moscas en desarrollo buscan otras condiciones, que son principalmente ambiente, floración, humedad, que haya agua, que estén presentes las condiciones ambientales favorables para que pueda continuar su desarrollo. Entonces, es la manera que se colocan estas trampas y tienen la funcionalidad de detectar oportunamente. Han cuestionado la presencia de trampas, que por qué no se da alguna presencia de trampas en sitios donde hay una infestación considerable. Bueno, el principio de esto es detectar oportunamente en zonas donde no hay presencia del gusano barrenador del ganado. Se están realizando investigaciones y se están realizando otras opciones, probando otras opciones de colocar trampas con cebos que sean tóxicos para la mosca y que puedan reducir también de esa manera la densidad de moscas que estén presentes en el campo. Estas trampas iban a ser colocadas en zonas de alta incidencia de casos y donde hay una alta concentración de moscas fértiles. Estas son las acciones básicamente que se han realizado, que se están realizando y obviamente pues tienen que seguir, obviamente la investigación tiene que continuar, se tiene que realizar. Es un problema que realmente pues es poco el tiempo que se tiene en México, obviamente pues una enfermedad exótica, un problema que no está presente, pues siempre causa este tipo de problemas. Hay un proceso de adaptación para poder implementar todas las estrategias que puedan pues ser efectivas para erradicar un problema, ¿verdad? Entonces se están realizando todas estas acciones. Además de eso, necesitamos de mucho apoyo de los colegas, de productores. Yo menciono aquí algunas funciones básicas o algunas acciones básicas que tenemos que aportar como médicos veterinarios en la lucha contra el gusano barrenador del ganado. ¿Y esto por qué? Porque los colegas son los principales ojos que podemos tener allá afuera, ¿sí? Porque ellos pueden detectar casos de manera temprana y eso es muy importante que al momento de detectarlos los reporten. Contribuyen de manera temprana a la contención si detectan casos y son notificados. Hemos tenido gran participación de los médicos veterinarios, no solo en unidades de producción pecuaria, sino también médicos veterinarios que se dedican a la clínica de pequeñas especies como perros y gatos y otras especies exóticas. Volvemos a repetir, es un problema que no solamente afecta al ganado, a los animales productivos, sino también a otras especies. Y el segundo lugar que tenemos en México como especie afectada son los perros. Y eso ha sido en gran medida gracias al reporte que hemos tenido de los colegas. Otra parte importante que tenemos, que tienen que hacer los médicos en el ejercicio libre de su profesión es la atención de casos. Si atienden casos con miasis, tienen que tratar la herida, obviamente colectar las larvas y notificar inmediatamente. Acciones de prevención y control como es el manejo integral, promover buenas prácticas de producción entre los ganaderos, acciones meramente de campo que pueden contribuir a disminuir la incidencia de casos, a evitar que más animales se infesten. Y eso tiene mucho que ver con las acciones que ellos tienen en el contacto con los productores, la capacitación y divulgación con productores en el momento de dar las asesorías, de atenderlos y además en el momento de compartir información. Uno de los retos y complicaciones que también tenemos es la desinformación que se da a través de redes sociales. Si se comparte esa mala información, eso complica todavía y desmotiva las acciones que se están realizando. Si desincentiva a los productores a notificar y a que se puedan realizar acciones de control. Y bueno, también la colaboración con las autoridades en las acciones que se realizan y en los operativos que se realizan para poder nosotros identificar nuevos casos y aplicar las medidas que son necesarias para tal acción. Por último, quiero dejar aquí un código QR en donde a través de la página de Senacica se encuentra toda la información importante relacionada con el gusano barrenador del ganado. Es información oficial, es información que se puede compartir perfectamente con cualquier persona, incluso con productores. Y bueno, es donde nosotros ocupamos el sitio para finalmente colocar toda la información que tiene que ver como entrevistas, como carteles, como información de trípticos. Toda la información que producimos se encuentra aquí de manera digital, incluyendo entrevistas de radio, televisión y algunas presentaciones, información relevante del gusano barrenador del ganado, en donde se puede consultar esta información oficial y que, bueno, pues obviamente puede ser compartida. Finalmente, dejo el número de emergencias en donde puede notificarse cualquier caso de miasis. Es un número que funciona a las 24 horas del día, los 365 días del año. También otro número que es a través de WhatsApp, donde se pueden enviar incluso imágenes. Y bueno, estos dos números, además de los otros medios de comunicación, son los que están permanentemente activos para cualquier notificación de casos de gusano barrenador del ganado. Con esta última transparencia estaría concluyendo mi presentación. Quedo atento a sus preguntas y agradezco la atención.
Thank you, Dr. Romero, for the excellent presentation and discuss the situation in Mexico and approaches that you are taking to control the screwworm infection. Unfortunately, we don't have time for questions because we are past the time. But thanks for sharing your contact information. So if anybody has any questions, they can be directed to you. And that concludes the afternoon session. And I will pass. And I thank the speakers for their presentations. And I will pass the microphone to Susana for the wrap up. Thank you.
Sí, gracias, doctora Luna. Excelente presentación, doctor Carlos. Desafortunadamente, por cuestión de tiempo, vamos retrasados, pero Bueno, no sé si ustedes están de acuerdo, creo que tenemos unos 5 minutos más. Podríamos tomar quizá 2 preguntas de los ponentes, bueno, de los participantes, para que puedan resolver algunas preguntas del doctor Carlos y la doctora Kim para atender este último tema de gusano barrenador. Entonces veo una mano levantada que es Mayra. Si quieres preguntar. Mayra, doctora Rahat, ¿puede formular su pregunta?
Yeah, sorry. Can I go or somebody else is going first?
You can go, Dr. Rahat.
Okay, thank you. It's a nice question from both Kim and Carlos, and I was wondering that other than wounds, is there any other hotspot or reservoir for these screwworm flies? Because I was thinking, what about that old school Bacillus thuringiensis biocontrol and the fungal biocontrols, if those can be applied in this situation, or is it just because the larvae are in the deep wound and some of those biocontrols are now possible?
No veo a la doctora Kim, pero no sé si puedes responder, Carlos.
Bueno, bueno, entendí que si además de las heridas, se puede presentar en otros sitios como orificios naturales y eso. Sí, es cierto, sí se puede presentar. De hecho, hemos encontrado algunos casos, principalmente en conductos auditivos, en vulvas de animales de hembras recién paridas, vacas principalmente. Son los sitios donde se han encontrado estas larvas. Realmente, la mayor parte de los casos que se han detectado están relacionados con heridas. Y todas estas tienen que ver con manejo, ¿sí? Y con prácticas que no se realizan de curación de las heridas y revisión constante de los animales. En cuanto a algunas otras medidas de biocontrol, se están investigando, se están realizando investigaciones al respecto para poder tener otras alternativas. En estos momentos no podría adelantarme o decir algo que ya se tenga como resultados, pero las alternativas se están buscando. Obviamente hay áreas que se están investigando y la intención es que estas áreas puedan tener, bueno, que estas investigaciones puedan tener un efecto en el campo y que pueda ayudar a los productores, porque de nada serviría una investigación que no pueda tener una aplicación en el campo. Entonces sí se están buscando opciones. Pues en breve tendremos información de estas acciones.
Gracias, Carlos. Hay otra pregunta en el chat que va a ser la última que vamos a tomar, de David Schneider. Él pregunta acerca de las trampas que se están utilizando, si estas trampas o si la inspección se realiza de forma visual o mediante PCR, o si quizá hay una oportunidad para realizar ahí o utilizar ahí inteligencia artificial.
Bueno, las trampas que se colocan son revisadas constantemente por los técnicos de los organismos auxiliares. Colectan las veletas, que ese es el tipo de trampa que se está utilizando, en donde se pegan las moscas, y estas veletas son enviadas al laboratorio central, al laboratorio de referencia, y los biólogos que obviamente realizan mediante la identificación taxonómica. No utilizamos técnicas moleculares para este caso, porque la identificación del parásito puede ser muy simple a través de la identificación taxonómica. Entonces, visual, ¿sí? Todas estas muestras son enviadas al laboratorio, los técnicos hacen lo propio, identifican las especies, las contabilizan, y hasta el momento no hemos identificado ninguna mosca de gusano barrenador del ganado. Las técnicas moleculares sí las tenemos, obviamente tenemos la infraestructura, pero definitivamente los costos se aumentan, y esto solamente se ocupa para corroborar alguna información, alguna muestra que definitivamente fuera muy dudosa, pero es muy raro. O sea, la mosca, las características taxonómicas tanto de la larva como de la mosca adulta son muy fáciles de identificar. Y obviamente, pues ellos tienen mucha experiencia en esto.
Vale, muchísimas gracias, Carlos. Desafortunadamente ahora sí estamos sobre el tiempo. Tendremos que cerrar nuestro taller. En lo personal, les quiero agradecer mucho a nuestros panelistas por su tiempo, por toda la información que nos otorgaron. Al inicio fue todo nuestro panel de influenza aviar, que como bien saben es una enfermedad emergente y con alto potencial pandémico, como lo vimos en las ponencias, ¿no? Que tiene una gran implicación para la salud pública. Y ahorita en la tarde hablamos del gusano barrenador, que es una enfermedad emergente en México y que bueno, pues, por su importancia transfronteriza también como grupo de trabajo de salud animal, consideramos importante hablar. En este aspecto, como bien lo mencionan, la educación y la comunicación es bastante importante, por eso se realizan todos estos talleres o estas pláticas para poder concientizar sobre el tema, ¿no? Entonces, nuevamente les agradezco mucho su tiempo, a los participantes también. Cualquier pregunta que tengan, lo pueden depositar en el chat, los ponentes lo van a tratar de contestar, y si no, ya saben, en nuestras redes sociales, en la página web de procinorte.net pueden encontrar más información o la información de los panelistas para que ustedes puedan contactarlos. No sé si doctor Roxanne, doctora Aruna, quieren decir unas últimas palabras.
Roxanne, you want to go first?
Sure, I can go quickly. I just wanted to say thank you for the excellent presentations both today and yesterday. I think this was incredibly helpful and insightful to me about the work that is going on between Canada, the US, and Mexico, and places where collaboration and rapid information sharing can allow us to respond much more quickly to disease incursions. So I really appreciate the organizers with AICA and keeping everything running smoothly, to all of the speakers and all the attendees for your participation. And I'll turn it over to Dr. Runa.
Thanks, Roxanne. Yeah, I have to say the same thing, and I thank all the participants. And this brings, and you can see the issues that we face as three countries, and the issues that the new issues that are coming up, and the limited resources, through the limited resources that we have, that we cannot address all the issues, and collaboration is the key. So this brings all the scientists together and address our questions and address the challenges, and get to a venue where we can collaborate with each other and use the funding opportunities available currently for even influenza, and I'm sure future opportunities will come for other areas that we have to deal with these new emerging diseases and emerging threats. And I want to thank thank AICA for putting this together. Thank you.
JC, ¿quieres decirnos unas palabras?
Gracias por su asistencia. It's you all three of you basically captured everything in terms of feedback today, and thanks to everyone, the whole team. Thanks to yourselves. Thanks to everyone participating. We've had over 300 participants over the 2 days, close to 350. So that's awesome. There's so much interest in these fields across the Americas, not just in North America. Do follow us on LinkedIn. Like I know Susanna had mentioned social media. So it's a brand new, the scientists had asked us to establish a LinkedIn space. So we've done that at ProCiNorte. For the grant, there's a grant section in the procinorte.net website for you to then download the form. Deadline is September 12th. If you're interested in bird flu and applying, has to be a minimum of 2 countries in terms of scientific collaborations. We also have a plant health workshop next week for 2 days, same format, kind of. And if you're interested in that, reach out. We're happy to share that with you. We look forward to working with you over the coming 12 months. Every year we come up with different themes, ideas. If you have some suggestions for the task force for ProCiNorte, reach out anytime. Happy to explore those ideas. And so thanks to Camino, who's put this all together in Mexico City for us as our coordinator. Reach out to her. She'll be posting it on in terms of the videos and presentations. It's always a question in the chat we get. I saw it again today. Well, that's a positive response. So everyone take care, stay safe, and we'll see you next time at the next workshop. Take care. Bye-bye now.
Gracias. Hasta luego.
Thank you.